Okay, good morning, guys. Sorry for the delay there. If you haven't already done so, and I notice most people have, just let me know in the comments that you're here. We'll get started very shortly. If you still have anything owing from Unit 1, and there are a few people in that boat, at this point, you really should be getting that into me. And you should also be contacting me to let me know what your timeline is to complete that stuff. So there are a couple people that are in that boat, which is probably not the best. Especially because we're about to tie up Unit 2 here. Okay, I don't see any questions, so my son was using my iPad. There we go. All right, so on day 7, which was Friday, we covered meiosis, the errors in meiosis, and then we were just going to get into the start of heredity, this idea of passing genes on to your offspring. Before we do that, we are going to do a quick review of the meiotic process. So like I said, if you have some materials, I'm just going to quickly set up a little table here so I can move some stuff around and you're able to see it on the camera. This coffee over here. Fit this up on my desk here. I think I can. Should probably get a different kind of camera set up for this, but all right. Let's do it like this. Move my mouse over here. Put my microphone on the other side so you have some chance of actually hearing me. Okay, so the organism that I'm going to simulate here is going to have a 2n number of 2. Okay, so this is an organism with 2 chromosomes. You're going to do the same. And keep in mind that that is the simplest eukaryotic configuration that you can get, because you have to have a homologue for every chromosome. So 2 is the lowest 2n number that you can get. If you only have 1 chromosome, then there's no homologue. And then for sexual reproduction, there's no way for that to work properly. So you grab my scissors here. Create my chromatids here. All right, so there they are. Keep in mind for this demonstration, showing the condensing of DNA and the decondensing of DNA, that's really hard to do here. So I'm not going to do that. I'm also not going to show the spindle fiber formation, or I'm mostly going to be focusing on what's happening to the DNA at every step. I'll talk about the other stuff, but it's just difficult to show here in my little desktop demonstration. You probably don't have the materials, so don't do that either. So imagine you have an organism here that has 2 chromatids inside of its nucleus, okay? Or it's chromatin at this point, okay? So 2 chromosomes. The distinction between those things is a little bit confusing, but a chromosome is one piece of unique genetic information, and the word unique is important there. Because if you make a copy of a chromosome, it's still the same number of chromosomes, because you don't double the chromosomes because it's not unique, because it's a copy. So here we have 2 unique pieces of DNA. We would say that there are 2 chromosomes in this organism. Okay, it's a 2n number of 2. This is a somatic cell, an adult cell. You could think of it as a spermatocyte or an oocyte because it's going to go through meiosis. Okay, well, maybe we'll just call it a spermatocyte for the sake of argument here, because we're going to just say it forms 4 sperm at the end. So we'll say it's a spermatocyte. That's a little bit simpler than the oocyte. Okay, and you could look at these chromosomes, and on them is contained some genetic information, obviously, some genes, and we could indicate on them, and you don't have to do this at home either, by the way. This is not integral to understanding the process, but I'm going to draw some alleles on these chromosomes. So they have genes. Genes are just, remember, a section that codes for a specific protein. And an allele is a version of the gene. So for example, you can have the gene for eye color, and you could have the brown version of the gene or the blue version of the gene. Those are called alleles, the versions. Okay, so let's say that we do have eye color, an eye color gene on here. It's going to be in the same spot or locus. The locus is the location, the spot on the chromosome. So let's say that there's a locus up here that has that gene, and I'm going to use B's. Okay, so brown, one of them has the allele for brown eye color, and the other one has the allele for blue eye color. So this is, I'm adding a little bit of extra information that sort of leads into our topic for today. Hopefully that's clear enough. I can't make this go any lower, but I don't think. Oh, wait, is there a magnification? Yes, there is. Hold on. Actually, I can make it go closer. No, what? 0.5. Oh, I thought it was going to go to 2. Okay, well, whatever. So you'll notice here that I've used a capital letter for one allele and a small letter for the other allele. So the reason for that is one of these is dominant and the other is recessive. And what that means is that the dominant, if you have a dominant allele, okay, a dominant version, it's always going to show, no matter what, over a recessive allele. So in this case, I said the brown one is capital B, and the blue one, oh, there's a big delay on this for some reason, and the blue one is little b. And so what that means is this person is going to be a brown-eyed individual, this organism, okay, whatever it is. Because the capital B, the dominant allele, is always going to show over the recessive allele. Okay, but you have 2 copies. And this, by the way, is true for every gene that you have. You have 2 copies, right? Because there's a homologous pair for every one of your chromosomes. You got one from your mom and one from your dad. The exception being sex chromosomes. So if you're a female, you do have a homologue for your X chromosome. Because you got an X from your mom and an X from your dad. But if you're a male, you got an X from your mom, but you got a Y from your dad. In which case, you're not going to have homologues because those are not homologous chromosomes. So I'm not sure if I mentioned this back when we were talking about the meiotic process, but the sex chromosomes don't actually do crossing over. They pair up as part of like the tetrad pairing because they still line up across the center together and stuff like that. They do all the other stuff, but the only thing that they don't do is crossing over. So the X chromosomes don't cross over if there's 2 X's, and the X and Y can't cross over because they don't have similar genes on them for X and Y. So the sex chromosomes don't participate in crossing over. All right. So here we are. We've got our 2 chromosomes. We got brown eye color on one, blue eye color on the other, but they're in the same location. Okay, I'm going to add another gene on here too. Let's say, what's another genetic trait? I mean, people always talk about eye color, but I want to pull something that's in the note today. Oh, I know. Widow's peak. So widow's peak is when your hairline comes to like a peak, a point at the front. I actually have one. It's kind of hard to see with my camera view here. But widow's peak is, I believe, dominant to having a straight hairline where the hairline just goes flat across. Okay, so I'm going to use T's for that. And I'm going to say that this person has widow's peak, capital T is for widow's peak, and little t means straight hairline. So if you were to think about what is this person going to express, what are they going to look like? Well, they're always going to show the dominant gene if they have the dominant gene. And so this person has the dominant gene for widow's peak, and so they're going to have a widow's peak. Okay? yeah, I guess there's really not much else to say about that. They will have a widow's peak. When we're talking about what they will express, what they will actually show, that the word for that is their phenotype. Okay, this is going to come up again today. I'm giving you a preview. And when we talk about which alleles they have inside of their genome, we call that their genotype. So the genotype that this person has for eye color is big B, little b. Big B, little b is their genotype. It's what alleles they have. Their phenotype for eye color is what they're going to show. So what they show here, the physical manifestation of this genotype, is brown eye color, because they're going to show the dominant. Same thing is true for here. We've got widow's peak and no widow's peak, but they're going to show the dominant. So their genotype would be big T, little t, but their phenotype, what they're going to physically show, is going to be widow's peak. All right. Now we got all that gene stuff out of the way. There's a little preview for what we're going to talk about today. This is interphase, okay? Anybody remember the key thing that happens during interphase for meiosis? Throw it out there. I mean, sure, you replicate your organelles and you make centrioles, but what is like the most important thing for our purposes? Remember, we primarily care about DNA here. Cool. DNA replication, synthesis. So I'm going to show that, and you show it as well. Keep in mind that what you should have in front of you right now are your 2 pieces of DNA. Okay, whatever colored object you used for that, because I'd like you to run through it with me with your hands. Okay, physically manipulate the things in front of you, and it'll help with memory and understanding. So you make a copy of your DNA. I'm going to do the same thing. So what I'm going to do here is put down another piece of paper here that is an identical copy to the original. I probably should have made these the same length to make this a little bit more accurate. I'll trim them down. I'll trim them down. That's actually pretty sad that I couldn't make them the same length. Let's try that again. Why make my metaphor more complicated when I don't need to? There we go. Now they're the same length. Cool. And you don't have any labels on yours, probably, unless you're using paper like I am. But just to be clear, when we make an identical copy, these are called sisters, okay? I'm going to have the exact same alleles on the copy because the copies are exactly the same. Now here, this chromosome and this chromosome are homologs to each other. They have the same genes on them, but you'll notice that if you look at the versions of the genes, they're not the same versions. Okay, some of them will be the same and some of them will be different, but in this case they're both different, the 2 genes that we've put on them. Okay, so they're homologs because they have the same genes, but they don't have the same alleles, the same version of the genes. But these are homologs to each other. Homologs have a similar function. All right, there's S phase. Now prophase is prophase 1. I better be as clear as possible here. Okay, so we're just doing interphase 1. Now in prophase 1, is the most complicated thing is going to happen here. We're going to get these 2 homologs to hug. Okay, so these are going to get together and hug. In my organism, in my sample organism here, I only have 2 chromosomes. But in your organism, the one that you're going to show me when you do your meiosis project, and we'll talk about this more shortly, you are going to have, I think, a minimum of 6 chromosomes, okay? So if you have 6 chromosomes, that means that you'll have to have at least 3 homologous pairs. So when you line them up across the middle here, there'll be 3 of these. 1, 2, 3. Okay? just keep that in mind. Or 4, potentially, if you do something that has 8. If you do something that has a 2n of 8. Okay? So, they've gotten together. This is I'm showing you the simplest version here, though. We'll keep it simple. So, they've gotten together in a tetrad. Okay? Now, once they're in their tetrad, crossing over is going to occur. So I'm going to show that by snipping off a gene from 2 of these between the homologs. Okay, homologs are what are going to do the exchanging. You don't exchange between sisters because sisters are identical, so that makes no sense. So we're going to switch here between 2 of the homologs. And it's an even switch. Okay. Same size piece, same piece, full genes. There's one. If you have a way to do this with your own, do it. Cut and use something to put back a piece. Okay? So if you look at the 4 chromatids, and remember that they condensed during prophase as well, so now they do look like sticks of DNA under the microscope. If you look at the 4 chromatids, you will see that there are no 2 that are identical. They are all different from one another. And again, that is obviously creating diversity, genetic diversity, in the potential offspring that you're going to create here, which is the purpose of this. So we've done a switcheroo. Now, that's the anaphase. Metaphase 1. These are going to line up. Now, I already kind of have them sitting lined up where I want them to line up. But why don't I, you know what, why don't I use my my little camera stand here as the metaphase plate. Okay? These are going to line up. They're still attached in their tetrad, but they line up so that one pair of sisters falls on one side of the metaphase plate and the other pair of sisters falls on the other side. And each one of them is going to do this. So and keep in mind that which side which pair of sisters goes on is random. So either you can have them sitting here like this, or they can switch, and the yellow ones here could be on the left and the blue ones on the right. So they could go like this, or they could go like this. And each pair, each homologous pair, each tetrad, will make that same choice, where one pair of sisters could go left or one pair of sisters could go right. And which way they go, left or right, is completely independent of the other decisions, which is why it's called independent assortment. They're each sorted independently and randomly. And then again, that adds genetic variation, because you're going to get a different mix of sisters in the left cell or in the right cell. Okay? So you got to make sure you talk about that during your presentation as well. That's another key component of this, because that's where variation occurs. All right, so now we have lined them up. Metaphase 1. Anaphase 1. The spindle fibers are going to yank on these and pull them apart. Okay? Keep in mind that the sisters are still together. Okay? They're going to be bonded together at their centromere. But now the homologs have been separated from each other. Okay? So the homologs are separated, but the sisters are together. And I had something I was going to say. I forgot. Okay, anyway, the sisters are together. Oh, this is what it is. Then we're going to get, okay, so that was anaphase 1. Now telophase 1, a new nucleus is going to form around each of these. And they're going to decondense. The DNA is going to decondense, go back into chromatin. And in cytokinesis, the metaphase plate is actually going to get pinched in, and then these are going to form two separate cells. Okay, so we have two separate cells now, one on the left here and one on the right. If you were to look at the amount of genetic information in each of these, at this point, you would say that each of the cells here is now 1N. They have haploid information in them instead of diploid. And the reason for that is that there is no homologous pair for each chromosome. Okay, their homologous pair went the other way and got put in the other cell. So each chromosome has to have a homologous pair in order for an organism to be diploid. But there are no homologous pairs here. There is slightly more than 1N information if you look at it, right? Because these are not identical anymore, so there's like an extra little bit. But it's not, you still wouldn't call this 2N. It's 1N plus a tiny bit. But you basically ignore that when you're talking about the ploidy. We call that the ploidy of the cells. Okay, we would call these haploid at this point. So they were diploid before, and now they're haploid. We would also say, by the way, that now each of these cells has 23 chromosomes in it. Even though, again, it's slightly more because there's a little bit of extra unique information here, but we kind of ignore that for the purposes of defining how many chromosomes are here. So we would call this 23 here now, and 23. Now, there are still 46 chromatids, because if you break these apart and actually count every stick, the chromatids are just the physical objects. So there is technically 46 chromatids here, but there's only 23 chromosomes, because chromosomes means unique pieces. Okay, and I realize that they are unique because of this, but again, we ignore that. So 23 chromosomes, 23 chromosomes. Okay, 1N. Now we're going to go through meiosis 2. So in interphase 2, people's gut instinct here is to say, well, you got to make a copy of these. Okay, don't do that. There is no S phase. Well, there is an S phase, but you don't copy the DNA during the S phase. Okay, it's basically a blank S phase. Temporaneously, like a time exists where that is occurring, but there is no replication of the DNA. So there is no S phase technically. There's no S phase action during meiosis 2. You do not replicate things twice. Okay, so you skip over that. You can pretty much think of interphase 2 as being nothing. I mean, you make new centrioles for cell division, and you make Golgi and whatever other parts just for maintaining the cell. But other than that, nothing. There's no DNA stuff that's happening. So that's interphase 2. Then prophase 2, you condense the DNA back into their sticks again. Remember, they decondensed in the last telophase. Okay, so they go back into their stick form. And so now they're visible under the microscope. And the nucleus dissolves. And that's about it. Okay, so this is way, way simpler than prophase 1. Okay, prophase 1's got the tetrads and the crossing over. And the second one is going to look a lot like mitosis. Okay, so it doesn't have any of that stuff. Now in metaphase 2, these are going to line up on a center line. Okay, in the cell. That's the metaphase plate. I don't really have anything to make a metaphase plate here, but you can imagine an invisible plate. And you know what I'm going to do? I'll make it a horizontal one so that they'll go top and bottom. Why don't I do that? Okay. The orientation doesn't really matter, but a metaphase plate now is going this way. And so we would, if there was more than one pair of sisters, they would get lined up along that metaphase plate, where one sister was on one side of the plate and one sister was on the other side of the plate. If I had some masking tape or something, I'd probably put a line on my desk, but I don't have any here. Okay. Now in anaphase 2, again, this looks very, very much like mitosis. You're going to have those spindle fibers yank on the chromatids and pull them apart from each other, okay, to opposite ends of the cell. And then telophase 2, these are going to get put into their own independent nuclei. They're going to decondense, and we're going to end up with, and then in cytokinesis 2, this is actually going to cut, and we'll end up with 4 different cells. If you look at the genomes of these 4 different cells, there's a couple things that you should note. First of all, they all have the haploid number of chromosomes in them. So remember that we were dealing with a 2n number of 2 in this example. So we started off with a somatic cell, an adult cell, with 2 chromosomes in it. But now when we look at our gametes, there are 4 different sperm here, each one only has 1. That's the haploid number, half of the diploid number. So if you start with 8 chromosomes, you need to end up with 4 in each gamete. If you start with 6, you need to end up with 3 in each gamete. Okay, so each has the haploid number. The other thing that should be obvious is if you look at the genetic information in each of the gametes, it's different. So every gamete has a different genome in it, a different combination of alleles. The genes are the same. Remember, they have to have the same genes. They have to have the same. They have to have one for eye color, one for insulin, one for whatever, if for a human. So it's all going to be all the same genes, but they have different alleles. The versions of the genes are different. Okay. I'm hoping after you have done this, hopefully you were doing with me, that was the idea. After you've done this on your desk, you feel a little bit more comfortable with this process. I realize that this is a complex process, probably the most complicated we do in this course, and that's one of the reasons why we spend time on it. But you guys are also going to do now, as a secondary thing, you're going to create a tool for teaching meiosis to somebody. Whoop. And we're going to go through what that project is going to entail now. Does anybody have any questions about what we just did together there? You've just done that and you're like, I don't know what's going on, or if that's how you feel, like you don't know what's going on at all, you should probably come drop by during the Google Meet, and we can absolutely go through the process again together. But if you have any questions about the process, I suppose we've already done together, but okay. I'm going to take a moment now and actually go through the assignment. So, because I want you guys to know what you're working on, and we have one more day basically to complete the unit. We're on day 8. Tomorrow is day 9. So we're going to be finishing off genetics tomorrow. We'll have our quiz. You can always take an extra day to write the quiz. And then we have one more day together, which would be Wednesday. And then you're going to switch into your other class. So we're actually going to start on the Wednesday, the first day of the animal systems unit. We're going to get into digestion. Man, I know this course moves very fast, but everything's going to be coming due soon, so we got to get on top of this stuff. So let's have a look at the assignment. So one thing to note for this assignment is that I've provided an exemplar. It's called Meiosis Live. What you are going to do is create a demonstration of meiosis. So very, very much the way that I just did. I just demonstrated meiosis to you and explained it to you. Now, I did it in a very basic way. I didn't include any title cards that showed you which step was happening at each step. So that's like an ultra basic version of showing it to you. But you're going to do the same thing, but a slightly more complex version of that. And the idea here is that you're gonna go through, show me all the steps, show me what happens in an organism that you choose. And it should be specific to your organism and specific to the sex of the organism that you choose. So you may pick a female yellow fever mosquito. Aedes aegypti, I believe, is the name of the yellow fever mosquito, which has, I think, 6 chromosomes. I could be wrong. I better double check that. But you're going to pick an organism that has a 2n number between 6 and 8. Okay? If you're looking for an organism, you can honestly just Google organisms by chromosome number. Easy. You have to pick a eukaryote. You can't pick prokaryotes for this because they don't do this. So, and they don't sexually reproduce, so they don't have meiosis. So you have to pick something that sexually reproduces. So the easiest thing to do is to pick an animal, to be perfectly honest. You can do plants, but I would really avoid plants because in actual reality, the meiosis in plants does look a little bit different. There are minor differences, and it's confusing. And as I did mention this in the other day, like some of them are actually tetraploid, which means that they have 4 of each chromosome instead of 2. They're not diploid. So I wouldn't go that route. I wouldn't pick a plant. So I would say stick to an animal, or you could do protists. It works with asexually reproducing protists as well. But don't pick asexually reproducing protists. Not all of them are truly diploid. So so you're going to design it around your organism, and by that I mean if you're going to be talking about a male, tell me where it's occurring in your animal. Is it, most of them have testicles or something like that if it's a male. Most females have something akin to ovaries, but I mean not every animal, but a lot of them do. So so you're going to discuss where it's happening. Okay, so that's what I mean by make it specific to your organism. If your organism has 6 chromosomes, you need to show me 6 in your visualization. Okay, that's the whole point of choosing one between 6 and 8. A number of people last quad that did this picked ones with 6 or 8 and then only showed me 2. I was like, well, what was the point of that? That's the whole point of picking something that has 6 or 8, is that you're going to show it with 6. So I would recommend color coding them the way I just did with the paper. It'll help you keep it straight, and it'll make it much easier for the viewer to understand, which is the entire purpose of this. So I highly recommend color coding them, although I guess it's not strictly necessary, but it will be very helpful. You need to name each stage. Okay, so ideally it would be shown somewhere in your stage on a little card or in the corner or wherever. You need to show each stage. So show what's happening at each stage and explain it. Certain processes you need to actually show happening. So if you're going to talk about crossing over, you actually have to show a crossing over event occurring. So make sure that visualization occurs somewhere in your explanation. Okay, like you actually show it happening. It should be obvious the difference between meiosis 1 and meiosis 2. I mean, they're very different from each other. So that's part of what I'm looking for here. You need to talk about crossing over and independent assortment specifically, and you need to talk about why they're important. Okay, so I'm looking for that. Remember that crossing over occurs in prophase 1 and independent assortment takes place in metaphase. Okay, metaphase, it technically happens in metaphase 1 and 2 because there is a choice in 2 as to which side the sisters are on. But because the sisters are largely the same, it's rarely talked about that it happens in metaphase 2. So I'm not really looking for you to describe that in metaphase 2. It doesn't really matter if you mention it in metaphase 2, but in metaphase 1 it's very significant because the homologs are very different from each other in terms of their actual alleles. So you do need to talk about it at least during metaphase 1. And then I already mentioned this, you need to explain where this meiosis is happening in your organism. Okay, so the idea here is that you are going to make some type of live presentation, again, very similar to what I've just done here. And there are lots of ways that you could potentially do this. You could create the easiest way to do this, in my opinion, is to make a Google slideshow and then just move the chromosomes around on the slideshow and show me what's going on, like as you're doing a screen record. So you could use Loom to screen record or Vidyard, or there's a number of services for screen recording. But you could just manipulate the chromosomes on your screen. I made a little video here of me doing that because I was like, oh, I better just like show what that might look like. So I very quickly just like throw together an example of how to draw some chromosomes in Google Slides and how to move them around and manipulate them and stuff. So you could do that. That's probably the easiest way to do it. And I'm not assessing you based on like how complex of a tool you use. Okay, you can use the simplest tool you want. You just have to make sure that everything is included and makes sense and is properly explained. I'm not looking for, is this a feature film quality presentation? I'm looking for, have you met the objectives of this? Okay, so that's one way you could do it. But it's like really wide open. I've had students make their own. You can do those interactive whiteboard presentations if you have a tool for that, where it sort of like records what you're drawing on a whiteboard. You've seen them on YouTube. You could actually do it on an actual whiteboard if you have a whiteboard at home, and then just film that. You could film it with a phone. You could film it with honestly, if you needed to, you could film with the webcam on your Chromebook. So you really don't need any kind of extra materials. Like I guarantee you have something that you can use to do this. Okay. If you feel like you're in a tight spot and you're not sure, let me know. Like I'll help you find some resources, but you definitely have what you need to show me this. So the key here is that the visualization component has to be made by you. So you can't just look up pictures of each of the steps and be like, there's this thing, there's this. The whole point is that you make the visualization. Okay, so there's no pictures from the internet in this at all. All right, I mean, other than you could show me a picture of your organism, but don't show me any of the steps of meiosis. This should all be your own work. Okay, it has to be correct for your organism, the correct number of chromosomes for your organism. That's the whole point. And you should make some attempt to show movement. Okay, what is going where and how is stuff moving? It doesn't need to be a complete animation, but somehow you need to show how things are moving, and because that's the entire point, right? This is all about taking DNA, copying it, and then moving it to the correct location. That's the whole purpose of making this as a video and not just, you could write an essay on this, but this is not as effective at explaining the process. So I want you to show the movement if you can. Okay, I mentioned color coordinating is great here. You don't have to do that, but it is very helpful to color coordinate your chromosomes. And as I mentioned, I have a little example there. So I also have an exemplar from a previous quad. I think this is from quad 2. So this, if you're looking for an example of a presentation that includes everything that you need and is well described, this is an excellent one. Okay, I know a lot of people are looking at this and being like, I have no idea what this is supposed to look like. Watch the exemplar. Okay, you don't have to watch the whole thing, but you really get a feel for how you present the process. Okay, so there's an example from a previous quad. I mean, again, that one's the next level. Like this person like made some animations and like did some. You do not have to do that. Okay, so it could be as simple as just having as I mentioned, just having a Google Slides where you're just manipulating stuff and recording your screen. That is perfectly fine. But this is just an example of one that has some extra stuff in it as well that you do not need to include. Okay, so that exists. I think I went through everything here. There's some ideas for some things that you can discuss. Some people have done claymation and stuff like that in the past. Again, not necessary. You guys can totally choose how you want to express this. Does anyone have any questions about the project, what it includes or what it's supposed to look like? I think it's fairly straightforward. I'm going to keep going. If questions pop up in the chat here, no problem. I will circle back, okay? What we're going to do now is we're going to begin discussing Mendelian inheritance. So there is a video down here that is sort of like an introduction to Mendelian inheritance. I'm going to get you to watch that after, okay, that we do the lesson together. And please note that the quiz, this is technically on day 7. I pushed it to day 8. It's not a big deal. But the exit quiz that's there is to go along with this content. So once you're done the introduction to heredity, that exit quiz is actually on day 7 if you're looking for it. We're gonna need part 3 of the course notes, which is right there. So if you haven't downloaded that already, you get it right there, and you're gonna open up in Cami, and we're gonna go through that together. So the diagrams have to include at least 4 chromosomes. So do you mean in meiosis 1 you should have at least 2 tetrads? That's correct, but it's actually 6 chromosomes, not 4. So you need to have 6 to 8 chromosomes. I shouldn't say 2 because you can't have 7, so it's going to be 6 or 8. So you need to find an organism that has 6 or 8 chromosomes to use. So your starting cell will have 6 or 8, and then in your final gametes that you produce at the end, you will have 3 or 4, depending if you had 6 or 8 to start. Exactly half that number would be in your gametes. You would have, if you had 6 total chromosomes, you'd have 3 tetrads. And if you had 8 chromosomes, you would have 4 tetrads, because the homologs are going to pair up. Hopefully I answered your question, Bree. Okay, so hopefully you guys are opening that up. Did all this. Nope, we did this already, not this. Nope. Where is it? There we go. Okay, so we already talked about this idea, which is that, so this is, all I did was just quickly note this here at the top of the note to sort of help us. We're going to do a little example together where we're going to assess our own genetics. So again, keep in mind that you have 2 chromosomes that have the same genes on them. They're homologs. And the version of the gene that you have is going to potentially be different on each homolog. So you could have a brown allele and a blue allele for eyes, for example. Allele just means version. Now, they don't have to be different. You could have the same allele on both. You could have brown and brown or blue and blue, but you could have brown and blue. Okay? So those are called alleles, versions of the gene that we already mentioned. I'm just bringing this up again because I'm going to use this terminology over and over while we do our little assessment of our own genetics together. So also, and I mentioned these terms already, but the combination of alleles that you have is called your genotype. And then remember that those alleles are going to be transcribed and translated. That's what happens in the in protein synthesis, right? That's what the DNA does. It's synthesized into proteins. That's going to lead to an outcome, something that actually shows up in the adult organism, and that is known as your phenotype. I like to always, whenever I say phenotype, in my head I always say phenotype, physical characteristics. Phenotype and physical have the same beginning, because the phenotype is the physical characteristic that you get from the genotype, or the genetic characteristic. So you have genetic characteristics that are in your genes, and then you have the physical characteristic, the phenotype, that is made from those genes. No questions about that? It's fairly straightforward. Okay, so a little bit more background information here. I mentioned that we use capital letters and small letters to denote dominant and recessive. So whenever there's an allele for something, or alleles for a gene, we show the dominant allele as a capital letter. And to be perfectly honest, the letters that we choose for these are basically random. You can pick any letter. Now, I highly recommend that when you are choosing these, and there are occasions where you get to choose the letter, I wouldn't pick a letter where the uppercase and the lowercase look the same, because that's going to make it very confusing for you later. So I would try and use letters where the uppercase and lowercase look different. Now, that being said, many times in the textbook they won't do that, which is super annoying. They use P's and things like that, that when you're not writing them on lined paper, small p and big P look very similar. Same things with like S's and things like that, capital S and small s. Like when it's not on a lined sheet of paper, like that's really hard to distinguish between capital S and small. So I would avoid that if you can. But the textbook doesn't, so so just deal with it, I guess. But even in some of these examples here, they did do it. So, and I didn't make this list. So, but anyway, dominant is shown with a capital, and recessive is shown with a small letter. So keep in mind that when we're talking about phenotypes, that remember that's the physical expression of these traits. Keep in mind that if you have two dominant, then you're going to show the dominant. If you have a dominant and a recessive, you're going to show the dominant. But if you have two recessives, then you show the recessive. So the only way to show the recessive trait, to actually have it in your phenotype, physically show it, is to have two recessives. All right, so I'm going to throw one more piece of terminology at you. This should all go in your terminology list, by the way. When both of your alleles are the same, and I know you know these prefixes from other science courses, but in science, homo as a prefix means the same, right? Like homogeneous mixture, it's all the same. So homozygous means that your two alleles are the same. If you are heterozygous, prefix hetero, like heterogeneous mixture, hetero just means different, and so heterozygous means that the two alleles are not the same. So you'll see this terminology come up a whole bunch. I will mention it. But homozygous just means they're both the same, and heterozygous means that they're different. Lots of fun terminology. Hey, right at the top. There's more coming. Yay. Okay, I'm going to keep going here. So what we're going to do right now is do a little bit of a analysis of our own genetics. We're going to look at some traits that are genetic, and we're going to identify, do you show the dominant trait or do you show the recessive trait? So all of the traits that we're going to be looking at now in the next minute here are traits that are known as Mendelian. Okay, named after Gregor Mendel. You're going to watch a little video about old Gregor when we're done this lesson. But Gregor Mendel is considered to be the father of genetics, and he really discovered that this was a thing, that you have two alleles for each trait, and it more applies to plants than it does to animals, really, because of the lower complexity in plant genetics. But there are very few traits actually in humans that are Mendelian. Mendelian means that they're single gene traits, which means you have one gene, and you can like point to it and be like, there it is. This is the gene that makes blank. This gene makes eye color. This gene makes hair color. This gene makes this. One gene equals one trait. That's called a single gene trait or a Mendelian trait. It's fairly common in plants for a lot of their traits to be single gene or Mendelian. Mendelian is the word that you'll hear over and over again. For example, and Mendel worked with peas, so I know lots of pea examples, but like, are the peas going to be round or shrivelly peas? That's one gene that decides that. Are they going to be green pea pods or yellow pea pods? That's one gene that decides that in peas. Are you going to have a tall pea plant or a short pea plant? That is a single gene trait in peas. For humans, most of our traits are way, way too complex to be controlled by one single gene. For example, human height. Human height is a mix of a whole bunch of different gene expressions for growth hormones, size of growth plates, bone size, bone density. I mean, you name it. It's probably like 50, 60 genes that are involved in expression of height. So it's not a single gene is equal to the trait. That would be Mendelian. So it's not Mendelian. I would say the vast, vast majority of human traits are not Mendelian. They're not one trait is made by a single gene. However, non-Mendelian traits, or polygenic traits is what they're called, poly meaning many gene traits, are very complicated to analyze. So we're not even going to talk about them in this course. We'll leave that for your university genetics course. We're only going to talk about Mendelian traits in this course. We have to keep things simple. It's just an introduction to genetics. So we're only going to talk about genetic traits. And you do, in fact, have a few traits that are Mendelian. Now, this isn't an exhaustive list that we're going to go through, but we are going to go through some of the human traits that are caused by a single gene. A great example, by the way, of something that is not controlled by a single gene is hair color. So hair color is actually not controlled by one single gene. So there's no brown allele or like blonde allele. It's not one gene. There's a whole bunch of different genes that contribute to hair color. And actually the same is true for eye color. It's not just one gene. Now there is a single gene that will determine whether you have dark eyes or high melanin production in your iris, or light eyes or low melanin production in your iris. It's not blue per se, but it's the difference between very dark and very light. Okay, so there is a single gene that does that, and we're going to talk about that gene in a second. That's a Mendelian trait. But the specific color that you get is polygenic, which means there's a whole bunch of other genes that decide, okay, you have light eye color, is that going to be gray or green or blue? That's the other genes add to that and make it specifically whatever your eye color will be. Okay, so something to keep in mind here. Humans are pretty complicated, and we don't have a lot of Mendelian traits, but we're going to talk about a few of them together now. So let's start with tongue rolling. Can you do this with your tongue? For real, though, can you do it? Check. And this is kind of silly, because what it's showing is that this person is using their cheeks to assist. You can't assist with your cheeks. Everybody can do it with their cheeks. Can you just stick your tongue out and roll it into a U shape without touching it with your cheeks from the side? I can't do it. So like I could honestly practice all day. I don't have the muscle that runs this way in my tongue that actually allows me to make this shape with my tongue. And that muscle, whether or not you have it, is a Mendelian trait. Okay. So tongue rolling is actually a dominant trait. They've decided to use R's here to denote that. So if you have the dominant trait, I'm going to go jump to the next page here. That would be your phenotype. Your phenotype is what you physically have. Okay? So under phenotype, I want you to record either you're a roller, you're a tongue roller, you can make a roll with your tongue, or you cannot. Okay? I cannot, so I'm not a roller. Because I'm not a roller, that means I'm showing the recessive trait. So the recessive trait is small r, and I know for sure that my genotype is little r. That's my genotype. If you can roll your tongue, it's a little bit more complicated, because either you have one tongue roller allele, that would be the first one here, Or you have both tongue roller alleles. You are homozygous dominant. Remember, homozygous just means they're the same. Okay? So that's if you are a tongue roller. You have one or the other. So another way to write that, you could potentially, for your possible genotypes, you could write this. Or another way to write that is big R and then blank. Blank just means that the second one is unknown. So you know your first allele is big R for sure, because you can roll your tongue, but you don't know if your second allele is a big R or a small r. There are ways to figure that out, genetic testing, and we're going to talk about something for dog breeders that they use to figure this stuff out, but in the moment, there's no way for you to know. So I want you to record now, what is your possible genotype? Mine is little r, little r. Normally, I would poll the class and say, okay, well, how much have it and how much don't or whatever. I'm not going to do that because it's going to take forever. So I tried doing it with the other class. It takes forever. So we won't worry about what percentage of the class has it here, but we're going to just observe ourselves. So second one, have a look at your ears. Okay? Do you have a dangler? Like I do? A little dangly guy at the bottom? Dangly earlobe? Or is your earlobe actually attached to your head? Is it an attached earlobe? That is a Mendelian trait in humans. One single gene decides that. So they've indicated that earlobes hang free, that is unattached, hang free, is dominant. Okay, and they're denoting that with a capital E. Okay, whereas little e would mean attached. Okay, so I'm unattached. I have free earlobe. That is my phenotype. My genotype, therefore, I have the dominant trait here, is going to be big E, but I don't know what the second one is. All I know is that I have at least one big E. Does anybody have any questions about this so far as we're going? Like how we're doing this? I'm gonna keep going, but if you do have a question, just type it into the chat, and I will get it as we go through here. Okay, I'm gonna keep going. I want you to take your hands right now and clasp them together like this. Okay, one hand over the other like this. Now I want you to look at your thumbs on the top. Is your right thumb on top, or is your left thumb on top? Have a look. Mine is left thumb on top. That is the dominant trait. Okay, so I've got left thumb on top, which means I have the dominant trait. And so I have capital C. Again, C's are not a great letter combination to use for these because big C and small c are hard to tell apart. But capital C and blank, because I don't know my second one because I have the dominant. If you have right thumb on top, you are recessive, and which means that you'll have little c here. Okay, next one. We'll keep this moving. Hold out your thumb like this and bend your thumb back as far as it'll go. Okay? Do you have a thumb that goes straight up and down? I'd have to push my thumb into that position. Essentially a straight thumb sticking up. Or do you have the thumb that goes back further than straight? Okay, significantly further than straight. Significantly further than straight is what's known as hitchhiker's thumb. Okay, but hitchhiker's thumb is actually the recessive. So the dominant here is to have a straight up and down thumb. And the letter they use for this is S's. Again, not a great letter combination to use. But I don't have a straight thumb. I've got the bent thumb, which is recessive, which means I've got little s, little s. But if you have a straight thumb, that would be big S blank. All right, next. These are all Mendelian traits. This is kind of fun. Widow's peak. I mentioned this one earlier. Pull up your hairline and see, like, does it come to a point in the middle? I do have a point right there. You can see my point, actually. I have a little point. My hair comes up to a point. That's called a widow's peak. And I believe that's the dominant. Yeah, the widow's peak is dominant. And they use W's. Not a great letter to use. But I have the dominant. So I have widow's peak, which means I have capital W blank. If you don't have a widow's peak, have a straight hairline, you'd be little w, little w. Dimple Chin. Have a look at your chin. Do you have a little dimple in it right here in the middle? If you're not sure what I mean, I can Google what a dimple chin is. You know what I'm talking about here. Dimple chin. Some people refer to it as a butt chin. It is this chin right here. I do not have that. So dimple chin, they're saying the cleft in the chin or dimple chin is dominant. Okay, I don't have a dimpled chin, so I am recessive, non-dimpled. Mid-digital hair. Okay, each one of your phalanges, your fingers, is made up of 3 bones. A proximal, a medial, that's the one in the middle, and a distal phalange, okay, bone. I want you to look at the middle one, okay, so the middle bone of each of your finger. I don't know if I'm indicating this properly on here. This middle, I gotta do on the camera side, the middle part, okay? Now look on the back of all of your middle digits, the middle part of each digit, okay? Do you have hair on any of the middle parts? I do. Actually, a bunch of it got singed off because we had a fire the other day, like a campfire. But it's still there. So that mid-digital hair is dominant. So, and they're using H's here for this one. So I do have mid-digital hair, which means I have capital H blank. If you do not have hair on any of your middle of your digits, the middle bone, then you would have little h. No mid-digital hair. Or hairless mid-digit, I guess you would call it. Okay, last one, and this one is a little bit more difficult to sort of pick out. But I mentioned that there is a Mendelian trait for more melanin in the iris or less. Okay, and so how you determine this is if you have darker colored eyes, that is, you would describe your eyes as green, brown, or black, you would say that you have the pigmented iris, okay? You have more melanin in your iris. If you have light eyes, like light gray or blue, you would call that light pigmentation. Okay? The dominant trait here is for dark. So if you have dark eyes, you are showing the dominant. That's green, brown, or black. I have brown eyes, and so I have dark iris pigment. So I have the dominant trait. If you have light eyes, light gray or blue, then you do not have this, and so you would have 2 small p's. Okay, so I have dark pigment, which means I have capital P. P's are very, again, not recommended because capital P and small p look very different. So I have dark pigment, but I don't actually know the second allele, so mine would actually be big P blank, because it could be big P, or big P, little p. Both of those would show as dark. Okay, so what we're going to do with this is we're going. Now this survey is a little bit old, so these traits may have shifted slightly in the population since this data was collected. I got this about. Actually, you know what? I didn't get it that long ago. I got it about 5 years ago, and it doesn't change that quickly. The percentage of these in the general population, but essentially, somebody has gone through and checked in a survey how many people have each of these traits, and has determined what are the most common phenotypes and what are the least common phenotypes in the general population. And this is an American survey, which is a mix of European and a whole bunch of different backgrounds. It's obviously different depending on where you're from, but the mix, the genetic mix in the US is fairly similar to what it is in Canada. And it wouldn't surprise me if, I mean, it's probably fairly similar in terms of how common it is here. So what you do is you start on the left and you pick the trait that you have, okay? So if you're a tongue roller, you would pick this, okay? If you're not a tongue roller, you'd pick the bottom one. I'm in the bottom one because I'm not a tongue roller, okay? That's actually the most common trait out of all of these is tongue rolling, okay? And then in the next one, if you are a tongue roller, you're going to use the top half. If you're not, you're going to use the bottom half. And then you're going to look at hand clasping. Again, is it left over right? That's capital C, or right over left. That would be little c, little c. And then you're going to go through and select your phenotypes as you go. Okay, so this is mine. I'm little r, big C, little s, big E, big P. Okay, and so I come out with the 21st most common phenotype. Okay, it's relatively rare, but it's not the most rare. So this is my, they call that phenotype number. It's just means in terms of being common, it's the 21st most common phenotype in the population. Phenotype number 1, which is that you have the dominant of all of these, is extremely common in the population. I usually have 2 or 3 people in every class that have phenotype number 1. As for phenotype number 32, which is the least common, which means you're recessive in all of these traits, I've never had that. In my entire teaching career, I've never had a class with somebody that has phenotype number 32. So it's obviously fairly rare in the population. It has never come up in, how long have I been teaching this course? 9 years. No one has ever had phenotype 32. So anybody now I'm going to ask, anybody in the class have phenotype 32? You're recessive for all of these traits? That would be fascinating. I'll have to make a note of it. Then I can always say I've only ever had one student that has phenotype 32. But so far it's never happened. Does anybody in the class have phenotype number 1? Can you guys do me a favor? Just in the chat, all I want you to do is type in the number that your phenotype is and just press enter. Just so people can get an idea of who has what phenotype. I think that would be fascinating. I mean, obviously you don't have to if you feel like this is too personal or whatever, but try it. I'm going to put in mine too. So I'm phenotype 21, so I'm going to put that in the chat. Now, generally, the numbers from 1 to 16 are much more common than the numbers from 17 to 32. Let's see if that holds true in here. I think it already does hold true, I think. Cool. I think we lost some people. I think. Or there's people out there that have no idea how to do this. If that's you, please say something. Fascinating. Fascinating. There we go. We got 2 people with 1, 3 people with phenotype number 1. There you go. That's pretty common. No 32s again. Interesting. Very interesting. I should also mention, as an aside here, that the dominant trait is not always more common than the recessive trait. Sometimes the recessive trait is more common than the dominant. They've just picked ones here where the dominant trait is more common than the recessive. That's all. So there's nothing to say that the dominant is also more common. There are definitely circumstances where the recessive trait is more common than the dominant trait. So those two things are unrelated to each other. It looks like they're related based on this, but they're not actually related. Ooh, we got a 31 though. Ooh, so close. So close. Okay, I'm going to keep going. Thank you for putting that in the chat. It's fascinating, interesting to see where people's phenotypes are. And as mentioned, we're only going to be talking about Mendelian traits in this course because it's just too difficult to assess polygenic traits. But it is possible to do. Just some advanced genetics techniques are going to be necessary for that stuff that you're going to learn in second year if you are a biology major. Oh, another 31. Ooh, that's exciting. We're so close. But you got the dark eyes, Molly. So close. I'm not sure if I mentioned this already. My son has blue eyes, and he is the only. Here, let's see if I can. Oh, do I not have my phone? Must have left it somewhere. Well, I'll get it later, but I'll show you. He's got bright blue eyes, and he's the only person in the family, except for my mom, that has blue eyes, which is interesting. Which means that my partner and I are actually big P, little p for that last one. So because we each gave our son the little p. So I gave our son the little p, and my wife also has dark eyes, and she gave our son the little p. And our son has blue eyes. Now, I know people are going to say, well, babies all have blue eyes. Yes, they do. That's true. Or most babies do. Because it takes a little while for the melanin to actually start being expressed in their iris. But he's 6 months old now. And normally by 6 months, you start to see some significant melanin production in the eye. They're like transitional eyes. They're eyes that are going to become a different color. But he doesn't have any transitioning happening in his eyes. They're just totally blue still. So he's probably going to still have blue eyes, which likely means that both of me and my partner, me and my wife, are both big P, little p, which is kind of cool. So I know what my genotype is, but only because of a special circumstance. Anyway. Let's talk about where we're headed with this. So, why do we care? Why are we even doing this? Well, we're going to be, for the next, normally I would say week, but now I'm going to say day, we're going to be studying heredity. The idea of how, it's the science of how genetic information is passed on and expressed in offspring. We'll talk about a few plant examples, but we're going to talk about several animal examples of this. And we're in general just going to talk about how things are passed on and expressed. Now, we're going to call on our knowledge of meiosis, which is how we have decided which alleles are going to go into our offspring. That's how we made our gametes. But we're going to be focusing on gene expression a lot more in this unit and talking about like how you go from having this in your genes to having this actual characteristic in offspring. So we were looking at just genetics in general, which is the science of genes and genetic variation in living things. And now we're going to be focusing in more on the heredity component, which is how you pass it on and express from parent to offspring. Okay, and we're going to be following Mendel's model. Remember that Mendel is mostly concerning himself with single gene inheritance or Mendelian traits. And so it's important to note that what we're going to talk about here, some of it does not apply to all heritable traits because those traits are not necessarily Mendelian. But we're going to start with Mendel. Okay. So I have a little bit of review stuff here. I'm talking about the fact that we have 46 chromosomes and there are 23 pairs, and then you have one from your mom and one from your dad. Normally, I go back and review this at this point in the course because we did, whatever, a week ago, but we did it like 2 days ago. We literally just covered this, so I'm not gonna go over this stuff again. We've already just done this. We already talked about this as well. This idea that there are places on each of your chromosomes or locations where particular traits or genes are found. Okay, for example, there is a locus here for hair color on chromosome 4, and there might be a locus over here for a particular genetic illness, or there may be a locus over here for making insulin on chromosome 9, or whatever. Okay? I believe that these ones are actually accurate. This is where they're actually found. But anyway, the thing is, we just did this. we were literally just talking about this. So I'm not going to spend too much time talking about this. But there's just the idea here is that there are actual physical places or loci where you find the alleles or versions of genes. Okay, or where you find the genes, and each gene has a particular version. You have 2 copies of each one, and so collectively, that is going to form the genotype that you have. Okay, you can be homozygous, which means they're the same, or you can be heterozygous, which means that the 2 are different. So really, what this is right here is just some terminology. I discussed this terminology, but I didn't write it down. So there you go. I wrote it down. I'll give you a second to get these. And I'm going to actually have a drink of water. And then we're going to start doing some problem solving. Changed my mind. I'm going to have some coffee. Stretch. All right. Hopefully that was long enough. I suppose you could pause, but okay, we already talked about this already. Let's see how we can trying to decide what to keep here. I'm always trying to slim these down. I've already slimmed it down quite a bit, but in order to keep time-wise, keep us on track. Because what do I want to cover here before we go away? Do I want to do this? What's all on the agenda today? Give me one sec, guys. Let's get rid of Dimple guy here. Oh, what a day. What a day. It's full. It's a full day. Yeah, we're definitely not going to get to the blood types thing. That we're going to have to save that for tomorrow, but we're going to have to do this part for sure. Squares, test crosses. I have till 10:20. Okay. Yep. I think we can do that. All right. That's reasonable. Okay. Let's talk about Mendel. So, one of the things that I think is a little bit confusing for people, and this just looks like a bunch of gobbledygook to you probably, is this idea of how are you going to pass a particular trait on to your children. Okay, so let's just look at a single trait. Keep in mind that obviously your chromosomes have a bunch of different traits on them, but let's not worry about all this. This is so we'll make it insanely complicated if we do this. I don't want to do that. We do in the normal course, in the non-hybrid version of this course, we look at inheriting multiple traits at a time. And there's like some different math that you can do if you're looking at, for example, instead of looking at what are the chances that your child will have brown eyes, we could look at what are the chances that your child will have brown eyes and brown hair, or brown eyes and widow's peak, or something like that. That's looking at multiple traits at a time. But it's a needless level of complexity here that we just simply don't need. So I'm gonna just get rid of that. So let's assume let's take a look at your genome, okay? And I'm gonna use widow's peak here. I'm gonna take it straight from the example. Okay, so let's say that you are heterozygous for widow's peak, and keep in mind that normally these would be sisters, right? So these would have been like this. Oops, it is. Why isn't this working? Oh, I'm gonna have to reset this, guys. I think this is gonna need to be reset. Let's save and exit here. Too slow. Okay. So, imagine you've got this genotype. Okay? You're big G, little g. All right? This is you. When you make gametes, you are going to make 4. Okay? And yes, if you're a female, only one of the 4 is going to stick and the other 3 are going to get tossed because there'll be polar bodies. But remember that you make more than one egg. You're going to make 1 per cycle, and it'll be in a different egg depending on the cycle. So really, like over time, these are going to be distributed evenly. But let's use sperm as an example here because I think it's a little easier to use the sperm example. So you're going to basically make 1 gamete. And actually, I previously had a crossing over event happen here. It's not necessary to do this, but sure. Let's say a crossing over event happens and then it looks like this. That doesn't really matter. But then when you're making gametes, you're going to end up with 1 gamete with this, 1 gamete with this, 1 gamete with this, and 1 gamete with this. So these would be your 4 sperm cells, right? Everybody with me on this? These are the 4 potential sperm that you would create. You'd have 1 big G, or a little g, or a big G, or a little g. You have 4 alleles, basically, in your genome for this gene. 2 of them are going to be paternal, and 2 of them are going to be maternal. So you could think of the 3 gametes as being the 4 gametes as being these possible ones. You got big G, little g, or big G, or little g. Okay? So if you were to mate with somebody that was also big G, little g, okay? So here we have a mating pair. This is you, or maybe you're female, doesn't really matter. And you're going to mate with somebody who is also a heterozygote. Heterozygote means that they're different, okay? It means you have the dominant and a recessive heterozygote. Okay, what that means is you are going to potentially provide big G or little g, and you don't need to show it twice, right? Because they're exactly doubled. That's like the same thing as saying this, okay, but like times 2. So you can, in other words, you can provide a big G or you can provide a little g to your offspring, and your partner can provide big G, or your partner can provide little g. Oops-a-daisy. Don't know how to draw G's anymore, apparently. Little g to their offspring. So when you bring these gametes together, there are only so many combinations of bringing these together and making specific combinations of the two parents, right? In other words, you could have the big G come together with the big G, or you could have the little g come together with the big G, or you could have the big G come together with the little g, or the little g come together with the little g, right? Those are the potential options that you can get there. So there are a number of different ways that you can combine together these parents. Well, there's a shorthand for this, okay? Because we need to assess this. We need to take a look at, okay, this is what the mom can offer. This is what the dad can offer. What are the possible combinations that we can make? So a tool that we use is this little guy right here. This is known as a Punnett Square, invented by Reginald C. Punnett. P-U-N-N-E-T-T, I believe. Punnett Square. Hope I spelled that right. I'm sure old Reginald is rolling over in his grave if I spelled it wrong. But the idea of the Punnett Square is that you're going to take the one contributor and put what their alleles are on the left, let's say, and the actual put it in on the left or the top doesn't really matter, okay? But in this case, we put the options for the male on the left, and we put the options for the female on top. Okay, so female on top, male on the left, and what we get in the middle here are the outcomes. Okay, I'm going to fill these in again so you can see where all this is coming from. So I'm just going to do the male's contribution to each potential zygote. Remember, a zygote is a fertilized egg. Okay, so all this is doing, this Punnett Square is doing, is summarizing how we can combine the female's alleles and the male's alleles together to make a baby. Okay, it's essentially looking at all the options that are available to a baby. Okay, so the male could give big G, okay, or the male could give little g, all right? And the female can give big G, so it could be a big G going with this big G, or it could be a big G going with the little g, or, and all I'm doing here is I'm lining them up, okay? So I'm lining up this one and this one. I'm bringing them across, okay? Or we could get the little g going along with the female big G, or we could get the female little g and the male little g. Okay, so all I'm doing is I'm bringing them together in the grid. All right, sort of just like if you ever done like a times table grid, you know, like 2 times 3, 4 times 6, or whatever. You know, you like bring them across in the grid, bring them together, and then these, what you get here at the end are the potential outcomes, the outcome genotypes for a potential baby from these two parents. Okay, so those are the parents, and here are the potential babies that you could produce. So a Punnett Square is just a tool that you can use to show what the potential options are for a baby from these two parents. All right, so we've got these options. Big G is going to be widow's peak, right? So we've got one widow's peak. Big G, little g also has a dominant, so it's going to be widow's peak. Big G, little g will be widow's peak, and little g will be a straight hairline. So if we look at these, we have 3, excuse me, 3 that are going to end up being widow's peak, and we have 1 that is going to end up being straight hairline if these two get together. So this actually tells us, because your chances of giving any particular allele to your kid is completely random. Remember, we talked about that independent assortment and that crossing over being completely a random process. So because they're totally random, your chances of getting any of these combinations in the end is equal. Okay, it could be any of these, and they're equally common because they're totally random. It's like rolling a dice and saying you have an equal chance of getting a 1 or a 6 or a 5 because it's just random which side you land on. So all of these are equal chances. So we can say that 75% of the time, these two people are going to make somebody with a widow's peak. And 75 is just 3 out of 4, right? 3 divided by 4 is 0.75. That's a 75% chance of making a widow's peak. And 25% of the chance, or 1 in 4, is going to make a straight hairline. And when we do this in real life, when we actually look at these percentage chances and how they play out in nature, this is true. We actually do see 75% of the time you get this, and 25% of the time you get this. And so the Punnett Square is a great tool for predicting your chances of having a particular trait in the offspring. Okay, and again, it's used for humans, but it's used for all kinds of other things. If you go to a genetics counselor, let's say that you have an unusual recessive trait in your family, an illness, a genetic illness, and there are lots of these. And then maybe your partner also has that genetic illness in their family. You would have your genome read, basically. They will genotype you in the lab. So they go through and read your DNA and figure out what sequences you have, which alleles you have. And so they see if you have the gene for whatever particular thing. And then they look at what your chances are of giving that particular allele to your children. And in some cases, people decide after seeing a genetic counselor not to have children, because if there is a 50% chance that you are going to have a child with a terrible genetic illness that would be perhaps really onerous, and there are some genetic illnesses that are horrible, horrific genetic illnesses. I mean, imagine that you have a child that grows normally till the age of 6, but then begins to cognitively decline and is going to die by the time they're 9, and maybe in horrible pain. That exists. There are genetic illnesses like that. And so it's easy to write this off as saying, well, sure, I'll just roll the dice, whatever. But I think it's probably because people don't realize what's actually out there. So when you have those kinds of things in your family, there's more to family planning, decision making than you might think. So, and people feel responsible for that because that information exists, and you are partially responsible for that. So, anyway, at least if you live in the developed world, those options often exist for you in terms of decision making. So. But that's just one example. So there's lots of other things you can use these predictively for. But if you're a genetic counselor, you use this information in real life. I mean, this is absolutely essential to figure out what your chances are of passing on a particular trait. So another way to say that is we can say that the ratio of genotypes, which is known as the genotypic ratio, is 1 to 2 to 1. In other words, 1 in 4 is going to be this genotype, 2 in 4 is going to be this genotype, and 1 in 4 is going to be this genotype. That's a 1 to 2 to 1 ratio of big G to big G little g to little g. That's called the genotypic ratio. It's the ratio of the different genotypes that you will get. Okay, and you can get that from reading a Punnett Square. You can also look at the phenotypic. If you're wondering where those genotypic ratios came from, I read it directly out of the table. So there is one big G. There are 2 big G little g's, and there is 1 little g in this table. So I just took it directly from the table. The phenotypic ratio, on the other hand, is when you assess what are the chances of having each phenotype. Remember, the phenotype is the physical manifestation of those genes. So we mentioned that 3 of those are actually widow's peak, I should use a different color there. 3 of those are widow's peak, and 1 of those, the little g, is straight hairline. So the phenotypic ratio are your chances, basically, of getting the phenotype, the actual outcome. That's 75% to 25% of widow's peak to straight. 3 out of 4 versus 1 out of 4. These are not the most complicated ratios in the world, but just keep that in mind. If you're not 100% sure what I mean by 75% or 25% or where I'm getting that, please say something. I will go through and show you the math, but I'm fairly confident that people are okay with the idea that 1 in 4 chance is 25%. 1 divided by 4 is 0.25. Hopefully it's not exploding anybody's brain. We're doing okay with that. If it is, though, just stop by. Don't worry. I mean, not everybody has the same background in mathematics, so that's totally fine. So Mendel did a bunch of experiments with this stuff. Okay, he grew some pea plants. We're going to look at one of Mendel's experiments in a second here. But he, through those experiments, figured out some basic things about peas. And, of course, these things are expandable to all organisms, but he didn't know that at the time. But anyway, the idea was, it was predicted at the time that this probably applied to other organisms. It does. So first of all, something that we've already discussed and that you know is one of Mendel's observations. By the way, these are the original tenets of genetics. This is sort of like just the origin of understanding genetics as a science and heredity as a science, or they're called Mendel's laws. But these are the tenets, or Mendel's laws. So the first one is that traits are controlled by a pair. Okay? You have 2 alleles for every gene. The second of Mendel's observations is the idea of the principle of dominance. And that is that some alleles, some versions of genes, are going to override the other allele. And so if you have the dominant allele, you only ever show the dominant. And if you have the recessive, it's going to show, but only if you just have recessive alleles, then it will show. And we've kind of already talked about that idea. Okay, so the idea here that we show, generally, when we write these, we show dominance with capital letters, and we show recessive with small. And recessive only shows up if both alleles are recessive. So that type of relationship where the dominant is shown, unless there are 2 recessives, you always show the dominant. That relationship is known as complete dominance. Okay? And as far as Mendel was concerned, everything was governed by complete dominance. Every type of inheritance had a dominant and a recessive, and that's because he was only experimenting with peas. So in actual real life, not everything is complete dominance. That's an oversimplification of inheritance. However, lots of traits are completely dominant. And we're going to talk about some complete dominant ones. We are also going to talk about some instances where it's not a complete dominance relationship. Okay, so just something to keep in mind. Not everything is complete dominance. So Mendel's second law here, or his second observation, this idea of principle of dominance, does not always apply. Okay, but it does in pea plants, but I'm going to show you some examples where complete dominance is not how things work. So, but it often is. And the last idea here is called the Law of Segregation, which is that alleles independently assort. We talked about independent assortment, the idea that they randomly go left or right during meiosis, okay, during the formation of sex cells, during anaphase 1 and 2. Okay, that's not actually where the sorting happens, but they separate during 1 and 2, anaphase 1 and 2. And what that means is that the chances of getting one parent allele or the other allele from the parent is completely random. That's all that the law of segregation really says, is that the chance of getting any particular allele in any particular situation is completely random. Okay, so in other words, if this is your setup here, if this is what you look like, your chances of giving big G to your kid or little g to your kid is a 50-50 flip a coin. Every single time, it's completely random. And that is because of independent assortment. It's totally random which way these are going to go in Anaphase 1 and Anaphase 2, or which way they're going to be organized in metaphase, really, is what determines that. But that's completely random. So that's the law of segregation. And again, it's important because if you want those numbers to play out in your Punnett Square, you want it to be the same chance of any of those potential options, big G, or big G, little g, or little g, little g. If you want it to be the same chance for each square, which it is, by the way, in reality, that's what, when you measure it, it actually is an even chance for each of them. The only way for that to work is for it to be totally random, which it is. Okay, so that's Mendel's last law. So how much time do I have? 10 minutes. Okay, I'm going to go through an example here of a genetics experiment that Mendel has done. And then I'm going to give you the opportunity to work on the questions for this part, which is page 193, number 5. You should also consider, while you're working on this, to try, once you're done, to try the practice quiz, which is on day 7 here at the bottom, this EXA quiz, and watch this little video. Okay, you may have to do some of this stuff during block 2, which is fine. I don't know how you guys organize your time during the day, but I'm assuming that everybody is using the break between 10:20 and 11:05 to break. Take a break. You don't have to. Obviously, you do whatever you want, but I'm certainly not going to assume that you're working during that time, because I'm not. So actually, I shouldn't say that. Sometimes I am. I'm often marking down here, but I got to stretch my legs too. Sitting in a chair is just like destroying my back. So for this long, I'm not used to sitting for so long. But anyway, let's talk about peas. Let's do a genetics experiment here. Okay, this is, by the way, not random. This is the exact genetic experiment that Mendel did in real life. He took some green pea pods and he took some yellow pea pods. I happen to already know which alleles are responsible for each of these and which one is dominant and recessive. Keep in mind that Mendel did not know this. Mendel had to figure this out. But I'm actually approaching this experiment here with hindsight. And so I already know this information, although Mendel had to work backwards and figure out which was dominant and which was recessive. He had to figure out the fact that there was such thing as dominant and recessive from this experiment. But again, I have perfect hindsight, and so I already know that green pea pods are the dominant trait, and yellow pea pods are the recessive trait. Okay? This little statement at the top explaining what each letter means is called let statement, and it is the first part of any genetics problem. You always explain what your letters mean. What is the capital letter? What does it mean? What is the small letter? What does it mean? And you know which one's dominant and recessive just by capital and small letter. Capitals are dominant and small letters are recessive. Okay? So you always start a genetics problem by explaining that, and those are called the let statements, just like they are in physics or whatever. You have let statements that say, okay, R is going to be the radius in this problem, and D is going to be displacement or whatever. Okay, so you do that in all mathematics, and it's similar here. So what Mendel did was he took what's called a purebred green plant. Purebred means that both of the alleles are the same. And again, Mendel didn't know that there were two alleles or anything like that, but what he did know was that if you breed green peas with green peas and get only green, and then breed them again and get only green, and breed them again and get only green, if they always make green under every single circumstance, then that must mean that they have only green genes in them. And again, he didn't know much more than that, but that's a purebred. A purebred means that no matter what you do, you always get green. Okay? And so he took plants like that were always breeding green. Same thing for the yellow ones. These are purebred yellow. They have the recessive trait, but they only have the recessive trait. I'm not really sure why I used green to highlight this up here. That's confusing. Should have made that yellow. Green and yellow. Okay, so these are the genotypes of the parents that he bred. And he didn't do this just once. He did it thousands of times. That's one of the handy things to doing genetic experiments on plants, is that you can do the experiment 1,000 times and then look at percentages and things like that, which is exactly what he did. So when you're crossing parents in a genetic experiment, we call that a P1 cross. P1 just refers to the generation. It's the first generation of parents. Okay, that's a P1 cross. So this is our P1 cross. This is the first generation of parents being crossed with each other, bred with each other. Okay? So the phenotype then of those parents is green and yellow. That would be the phenotype of the parents. These are all things you're going to include in your genetics problems. I'm going to summarize that later. So what you have to do to figure out what happens in this circumstance, and again, I know this because I have the benefit of hindsight. Mendel had to figure this out, so he actually worked from the results backwards. But I'm working forwards because I get to know all of this. I know how this works already. So you take the one parent and you put it on the top. It doesn't matter which one goes up there, and then you put the other parent on the side, and then you're going to run through your Punnett Square. So we're going to get big R, little r, big R, little r, big R, little r, big R, little r when we combine these two together, because each parent can only give either a little r, or if you're the other parent, you can only give a big R because you're homozygous. Okay, so when we look at the genotypic ratios and the phenotypic ratios for this, well, all of the genotypes for the babies are the same genotype. Look at that. Every single one of them is big R, little r. And because big R, green, is dominant, the phenotypic ratios, that's what they will physically look like, is 100% green. So, whoa, what a weird observation to make as a scientist. You take something that always makes green, and you take something that always makes yellow no matter what, and you breed them together, and you get always green. Every single time you breed these two together, you get 100% green. So that's weird. I mean, immediately you have to be thinking there is something about the green that is like stronger than the yellow, because when you put green and yellow together, you always get green every single time. So this starts like the wheels turning in Mendel's mind. He's like, oh, something is going on here. What a weird observation to make. And a lot of stuff in science works like that, where you make a strange observation, and then that's where you actually figure out how things work. So Mendel was like, all right, that's a weird thing to see. I mean, we know the reason why. Again, we know Mendel's laws. He didn't know this at the time. Okay, so what he did then was he said, okay, I need more information here. I'm going to take the babies from this first union here. So the big R, little r plants. He assumed that they were all the same. They were, in fact. So good on him. And he said, I'm going to cross them with each other. Okay, so when you take the babies of a union and you cross the babies with each other, that's called an F1 cross. Okay, F is for filial. Filial are the offspring of the first cross. Okay, and F1 just means the first generation of fillies, of offspring from the first. And the word filly obviously is a reference to horses, because after this point, these principles were, there wasn't called that back then, but these principles were applied to horse breeding, often applied to horse breeding, which is why a lot of the terminology here actually comes from horse breeding. But the F1 cross is the first filial generation crossing. Okay, first filly means the first, I think I wrote filial above there. Yeah, there it is, first filial. Okay, so it's basically taking the babies from the first union and crossing them. So now we're taking big R, little r, and crossing it with big R, little r. Both of those are green, right? They both have the dominant allele, and so they're both going to be green. So we're crossing green and green, but very specifically here, we're crossing the first generation of the first cross, okay, because there's something different about them. So when you do that, what happens is, and so we put one on the top and one on the left, and we figure out what they're going to be. So we get one big R, two big R, little r's, and two little r's. And again, I know that. I can do this Punnett Square. Mendel didn't have this. But what Mendel ended up seeing, he didn't see the genotypic ratios because you can't see that. You can only see the phenotypes. But we know that 1 of 4 is going to be big R, 2 of 4 is going to be big R, little r, and 1 of 4 is going to be little r. So another way to write that is the genotypic ratio of 1 to 2 to 1 of big R to big R, little r to little r. Okay, I'm just transferring this directly over from the table over here. Okay, so there's 1 big R, there's 2 big R, little r's, and 1 little r. What Mendel did see was the phenotypic ratios. So what he saw in the end was that 3 of these, and remember, anytime you have a big R, you're going to be green. So 3 out of 4 were green. This is the phenotypic ratio. And 1 out of the 4 were yellow. So you write that as 3 to 1, green to yellow, or 75% green and 25% yellow. So that is what Mendel actually saw. He saw the phenotypic ratios, the ratios of green peas to yellow peas. And he didn't only do this with pea color, by the way. So it started with a pea color experiment, but then there were other traits that you can use in pea plants. He used wrinkly and round peas. There's other Mendelian traits here, or single gene traits in peas. And so he did this with a whole bunch of different traits, and he got the exact same result every time, that when you take the first filial generation, 3 out of 4 pea plants are going to show the dominant, and 1 out of 4 are going to show the recessive. It's almost exactly 1 out of 4, okay, which is really cool. And obviously, the higher number that you do, the more likely you are to see that. And he did it with thousands. And so when you actually look at it on paper, it ends up being 3 out of 4, basically, every time you do it. So Mendel looked at this and was like, oh, my goodness, this is such a weird thing. Why would I get this? And from these end results, he actually worked backward and guessed that this is what must actually be happening, that there must be 2 alleles for each trait, and that in order to get these results, this is what actually must be happening on a genetic level. We must be seeing this arrangement of alleles. And so then he designed a number of other experiments to test this, to see if that is in fact what is happening. And it predicted the results every single time. That's cool. So when that happens in science, when you make a prediction based on your observations, and then you test it, and it's confirmed to be true every single time you test, that's what's known as a theory. That is a scientific theory. Okay, and so this is our original theory of genetics. It holds up pretty well. So, hooray, came from growing pea plants. I'm going to leave it there. I ran out of time, obviously. And so if you want, again, you can do whatever during your break. There'll be some time to work on this in the second half, but you guys are going to do number 5 on page 193. We got to squeeze this video in at some point. It's simply a review of Mendel. Talks a little bit about Mendelian traits and pea plants and stuff like that, in a little bit more detail. And then there's a little exit quiz for you to try. What we're going to get to in the second half today is we're going to talk about some situations where, well, you're going to do some genetic problem solving. We're going to talk about how to determine whether something is a purebred or not in practice. And we're going to look at this idea of incomplete and codominance, which are other inheritance patterns that are not complete dominance. Okay, and now I'm going over time, and I'm going to give you a break. So, again, you can start that at your leisure. Enjoy your break. I will see you guys shortly at 11:05. Oh, question. Do you have to do page 193? Question 5 on page 193. Yeah, that's this one. Yeah, you're going to do that question. Yeah. You can do it now, or you can do it later when you're working on stuff. Totally up to you. But I would take this break. That's just me. But you're just going to work on it later. So if you want to do it now and then not do it later, that's fine too. Yep. It says on the bottom of the. It also says it, by the way, on Brightspace. Complete question 5 on page 193. Anything that's in the notes that you need to do is written on Brightspace as well. I always put it on Brightspace as a separate step as well. So if you're not sure, you can always look on the day on Brightspace and see whether it's there. If it's there, you do it, and if it's not there, you don't do it. So it's always there. I double check. So just as a heads up, you can always look on Brightspace, and there it is on Brightspace. So okay, enjoy your break, guys. All right. Please let me know if you are here for the second half in the comments. Okay, let's talk a little bit more about types of inheritance. So we looked at some of Mendel's science, the original experiment that there he is. Everybody's here. We looked at the original experiment that led to some of these conclusions about genetics and figuring out that we had 2 copies of each allele, which is pretty cool. So I mentioned previously that this form of inheritance, this idea that if there's 2 alleles, one of them is dominant over the other, and the dominant one always shows if it's present, and if you have the recessive, it only shows if you have 2 recessive. So that inheritance pattern is called complete dominance, and that all the traits that Mendel was working with exhibit complete dominance. Lucky for him, it would have been very confusing if they didn't, because he would have got different results then for his experiments, which probably would have prevented him from coming to such clear conclusions. So anyway, Mendel was working with traits that were complete dominance. All of the traits that we were examining, the human Mendelian traits with chin cleft and hair thing, what's it called? Widow's peak, attached earlobe, dark or light colored eyes. All of those things are traits that exhibit complete dominance. So it makes it a little bit easier to analyze. We're going to look at a couple traits here. I'm going to give you some example genetics problems that are solved using complete dominance, or that follow the inheritance pattern of complete dominance. And so we're gonna run through a couple of these together, and then we're going to look at a strategy for determining whether or not an organism is a purebred. That is, they have, and typically purebreds are homozygous dominant. So we're going to be looking at this idea of, is an organism homozygous dominant? You've probably heard the word purebred used before with regards to dogs. It's used in breeding of all kinds, because purebred is usually what you're looking for as a breeder, because you want all of the dogs that you breed with that dog to have the same traits. So breeding a homozygous dominant animal is usually essential to doing that. So anyway, we're going to talk about how to determine that and some strategies for figuring that out. And then we're going to talk about 2 different variations on complete dominance. So complete dominance is not the only inheritance pattern. We're actually going to talk about 3 other inheritance patterns that are not complete dominance, and remember that complete dominance means that there's a dominant and a recessive allele. So it doesn't always work like that. The 2 that we're going to talk about today are incomplete dominance and codominance, and both of them sort of have their own signature pattern of inheritance. And tomorrow, at the beginning of tomorrow, our last topic for inheritance is that we're going to talk about the idea of multiple allelic inheritance. And so far, everything that we've talked about and everything that we will talk about today only has 2 alleles. So you can either have this or this. You can have brown hair or blonde hair. You can have little a or big A. You can have little t or big T. But what happens when there are 3 alleles? Obviously, you can't use little a and big A to represent them anymore because there's more than just 2. So obviously, there is a whole other strategy involved then in when there are more than 2 possible alleles. And you guys know of circumstances where this is true. Like there are traits where there's more than 2 options, for example. We mentioned dark or light eye color. That's sort of a 2-option thing where it's either dark or light. But when we're talking about specific color, of the eyes, there's far more than just 2 options. And so not only is eye color polygenic in that more than one gene is involved, but in those genes themselves, there's more than 2 versions that you can get. So there's more than just big A and small a. And then we start getting into a little bit more confusing territory. And that's what we're going to examine tomorrow. We're going to look at a relatively simple example of multiple allelic inheritance, where there's more than 2 potential options for alleles. And then we're going to look at blood types specifically, which is an excellent example of that. So anyway, that's where we're headed. So I'm going to start here by going through an example problem here of using a Punnett Square to solve a genetics problem. We already kind of did that. We looked at the Mendel example, but I want to show you what you need to include in your problem solving when you solve a genetics problem. So a typical question is going to look like this. In humans, brown eyes are dominant over blue eyes. So first of all, the relationship is going to be stated. In this case, it's saying brown is dominant over blue. Now keep in mind that when you're reading problems in the textbook or reading my problems that I'm giving you, they're not always real. Okay? I mean, they're a real problem that I'm giving you, but keep in mind that the traits that are being given and the are not always actually Mendelian in real life, and they don't always operate quite so simply in real life. So this is an example of this. Brown eyes are dominant over blue. That doesn't exist. So that's not like a real thing in humans. We did talk about the idea of light eyes and dark eyes being a real Mendelian trait, and that is real, but this one isn't. So just something to keep in mind. That table that we did of Mendelian traits in humans, those are all real. But a lot of the stuff that's used for examples here are made up examples of alleles. And the reason that they're made up is because there aren't that many actual Mendelian traits in humans. So just to prevent me from repeating the same examples over and over again for everything that we do, I've made some up. The textbook does it too all the time. They just make stuff up. So again, the problem solving is the key here, not the fact that these are true facts about what is and is not Mendelian. Just as a heads up, because people always ask, oh, I thought that wasn't a Mendelian trait in humans. It's not. Anyway, brown eyes are dominant over blue. So you're going to be told that. Or you'll be able to pick up which one's dominant from the way that the question is presented. So sometimes the way that the question is presented, it's obvious that the heterozygote, remember the heterozygote is when you have a capital letter and a small letter, when you have a combination of dominant and recessive, will be blank, this trait. Well, if you know that the heterozygote is blank, then you know what the dominant is, because that's the dominant, right? The dominant is going to show in a heterozygote. So anyway, in humans, brown eyes are dominant over blue. If a homozygous brown-eyed man is crossed with a blue-eyed woman, what are the expected genotypic and phenotypic ratios of the F1 generation? Remember that F1 means first filial. It is the children of the P1, the children of the parent generation. Okay, and the reason that you ask about ratios is that's really like asking what is the percentage chance of whatever outcome. Okay, that's basically asking the same question as that. So when you are answering these questions, the first thing that you always need to include are let statements. So I have them labeled there as number 1. I labeled the steps over on the side in blue. You pretty much always need to include these. Way back in the day, now it feels like forever ago, when we wrote the test for this unit and you wrote these out, you went step by step and, okay, you get a mark for showing me number 1, number 2, and number 3 for using the problem solving technique. The problem with that is very difficult for me to get from you and assess. I tried doing something like that earlier in the year, and it was a complete failure. It was too complicated. People were taking photos of things, and things weren't clear. And so I stopped doing that, and I switched to an all multiple choice model, which basically you solve the problem, but then I ask questions about basically what you got, and then I use that to assess whether or not you use the correct formatting in the problem. So I sort of reformatted how I assessed it, just for expediency. You still, I mean, it works out to be the same, but when you're doing these practice questions, it would be a good idea to write out the full answer set the entire time. Because just like when you use the grass method in physics or whatever other discipline you use grass method for, physics often, chemistry as well. The problem solving technique is part of what you're learning, because the more complicated the problem gets, the more important it is that you have mastered the technique, because you can sort of rely on that to make your problem solving work easier. Okay, it's if you don't use the technique, you may struggle at points to figure out what to do next in a given problem, and this helps with that. It helps keep everything flowing. So I recommend that you all follow these steps every time I ask you about these things on the quiz, like what would be the phenotype of the parents or whatever in this particular problem. You basically have to write them out anyway. So, but when you're doing these homework problems, you should write out the entire solution. That's the whole point. I know I already mentioned this previously, or I'm pretty sure that I did, but if you are writing out full problem sets, and you want to include, if you're doing it on paper, you can always just take a picture of it and insert the picture into your homework document, if it is textbook work, and there will be some textbook work here. Okay, so first of all, you start with the let statement. So you got to tell me what is the letter that you've used for the dominant, and it should be capital, and what is the letter that you've used for the recessive trait, and it should be lowercase. You should choose letters where the uppercase and the lowercase letter do not look the same or similar. So don't use S's, don't use P's. This is I suppose, a personal preference on my part, but realistically, like, you're shooting yourself in the foot if you do it the other way. You're going to confuse yourself. So, and I ask a question about this on the quiz as well, but don't make them letters that are easily confused. It's not a good idea. So I said let capital B be brown-eyed allele and little b be the allele for blue eyes. Okay, it's evident in which one is capitalized and which one is lowercase, which one is dominant. So the dominant is always going to be the capital letter. So then I've just mentioned here, we're going to cross the parent generation to get the first filial, the F1 generation. You don't need to include that. You don't need to say that, but that is what we are doing here. The second thing that you do need to include is you need to tell me the genotypes of the parents. So you'll remember from the question here, it says a homozygous brown-eyed man. Homozygous brown would be big B, big B. And then it says a blue-eyed woman. Okay, blue-eyed is the recessive trait. So the only way to get a blue-eyed woman is this. You have to have 2 recessive to get blue eyes. Okay, so there is a little bit of inference that you have to make here in order to determine that. But this should be fairly obvious that the woman has to be little b, because that's the only way to get blue eyes. You have to have only recessive. Okay, so those are the genotypes of the parents. Then the third part is you have to tell me the phenotypes of the parents. Now the order of this doesn't really matter. If you want to tell me the phenotypes first and then the genotypes, that's also fine. This is very similar to in physics when you write your givens. Okay, you're basically telling me what you know already from looking at the information in the problem. This one's a very straightforward problem, but sometimes it takes a little bit of thought to figure out exactly what it is that you have been given. Okay, but these are the givens. You've been given this information. Then step number 4 is you're going to do a Punnett square. Where's my Punnett square? There it is. I did it over on the side here. Okay, so I put my one parent on the top and my one parent on the left. So we got big B, and little b, and then I did my cross. That's how I'm going to get my F1 generation, which are the ones in the middle. Okay, so in this particular case, they're all big B, little b. So when I do my genotypic ratios, they're pretty straightforward here. It's 100% big B, little b. You can't really do a ratio per se because it's not 1 to anything else. It's 100% of one thing, so it's easier here to just write it as a percent. So it's 100% big B, little b. For the phenotype, same thing. Well, they're all the same, and they're all going to be brown. So the phenotypic ratio, which is the last step that you need to include, is that they're all brown eyes. Now, I did write a 7th step here at the bottom, which is typically you answer the question, okay, which is what are the phenotypic and genotypic ratios. And I just wrote that in. I said, therefore, the genotypic ratios of the F1 generation are 100% this, and the phenotypic ratios are 100% this. So the step 7 is to just answer the question. Now, in this particular instance, answering the question is just writing the previous 2 steps again. So, yeah, it could be argued that step 7 is not particularly important here. I don't disagree with that. But there are going to be some questions that they're going to ask you to analyze the information that you've just discovered here about this genetics problem, and ask you a question that isn't just tell me what was step 5 and 6, in which case it becomes a lot more important to include step 7. So I would just get in the habit of including a therefore statement at the end. This is the GRASS method for genetics problems. That's what it is. Okay, so typically you include a therefore statement at the end. As a additional question here, I'm just going to run through this again, but I'm going to do an F1 cross. Okay, so an F1 cross, remember, is when you take the babies from your first cross here, okay, the first filial generation, and you cross them with each other. Now that's going to give you your F2 generation. F2 just means what happens when you cross F1. When you cross the first babies, F1 generation, the F2 generation is the babies you get from the babies. It's the babies' babies. Okay? So, we're going to do a cross the first filial generation to get the second filial generation, which is written as F2. This is not in the initial problem. It does not ask you to solve for this. I'm adding this on, okay, in case you're asked to solve the second filial generation. Now, in this case, you've already been given therefore statements, you've been given let statements, because you gave them already, right? You already defined what B means, big B and small b, so you don't need to do it twice in the case where you're doing the second filial generation. In this case, you can just jump straight to number 2, which is what are the genotypes of the parents? And so you look up, well, what are the genotypes of the parents? Well, they're all going to be big B, little b. All right, so this is a big B cross with a big B little b crossed with a big B little b. And the phenotypes of the parents are brown. So they're going to be brown crossing brown. Then we do our Punnett Square. I put it off to the right here, but keep in mind that you can place this wherever you want on your page. Right below is fine too. I'm just doing this to save space. And these are the results you're going to get here if you cross these two. So remember that one parent goes on the top and one goes on the left. So we've got one big B, 2 big B little b's, and 1 little b little b. So our genotypic ratios, which is step 5, is 1 to 2 to 1 of big B to big B little b to little b little b. Those are the genotypic ratios. And the phenotypic ratios are 3 brown to 1 blue, because big B is going to be brown, big B is going to be brown, but little b is going to be blue. So in this case, we have 3 brown-eyed children for every 1-eyed child. So that is a 75% chance to be brown-eyed and a 25% chance to be blue-eyed. And then that's the conclusion here. So in the F2, 75% of the offspring will have brown and 25% will have blue. Does anybody have any questions about sort of like the general procession here of how you solve these problems? It works pretty much the same way every time. You're starting with the parents, you tell me their genotypes and then their phenotypes, you do your Punnett Square, you look at the genotypic ratios in the offspring and the phenotypic ratios in the offspring, and then you answer the question. Fairly straightforward. We're going to use this for all of the problem solving. Now keep in mind that most of the questions proceed like this. However, in some of the homework questions, what they're actually going to do is they're going to give you the offspring. So this is a little bit different in that they're going to tell you, well, these are the babies that will be produced in this particular union, and this is not as straightforward. Okay, so this is not like a 1, 2, 3, 4 step 1, 2, 3, 4, 5, 6, 7. This takes a little bit of thinking outside the box. They may give you just thinking inside the box, thinking inside the Punnett Square anyway. They're going to give you the offspring. They're going to say, well, there is a little b being produced in this union. And here is one of the parents. One of the parents has this. What does the other parent have? And so here you're putting it into the Punnett Square and saying, okay, well, one of the parents is this. And then you're looking up at the top and saying, okay, well, what does the parent have to be in order to produce that combination of alleles? Okay, so some questions are going to ask you to work backwards, and that's fine. And that doesn't follow the same sort of 1 to 7 procession, and that's also fine. Okay, so sometimes you're not going to be including all of that information, but the typical genetics problem is going to proceed like that. Filial means basically baby. First offspring generation. And the reason that the word filial is used is because a filly is a baby horse. And because these this way of analyzing genetics was often used in horse breeding. That was one of its original functions, especially in animals. And so the term filial became synonymous with the offspring or the children of the parents of the first breeding pair. Okay, so we call the first breeding the first offspring. So in this case, if this would be the F1 generation right here, these big B little b's, they would be the first offspring generation from these parents. We call that the first filial generation, or the F1. Why fillies? Again, it's because of the horse breeding connection, and we just stuck with that word for some reason. Biologists, I don't know. They could have called it something else, but yeah, it's because of its association with horse breeding. So that's the first filial. When we talk about the second filial generation, that means the second generation of babies. And it's usually made from the first generation of babies. So you take the first generation of babies and then you cross them, and then that's how you get the F2 generation. The second filial generation is going to look like this. It's the offspring of the babies. Hopefully that answered your question, Ivana. But that's what filial means. Okay. Now I want to talk about a problem that comes up all the time for breeders. And that is, do I have a purebred? That is an important question you have to ask, because if you want to use an animal for breeding purposes, you have to know that every time you use it, it's only going to give specific alleles to the babies. Now, you don't care about every single allele in the entire genome. You only care about the characteristic alleles. So if you look up Chihuahua characteristics or German Shepherd characteristics, there are specific characteristics that are attributed to that breed. So German Shepherds have a certain shaped face, or I don't know what this is called in a dog, snout, I guess. They have a certain shaped ear. They have certain coloration. These are all genetic traits. And those are characteristic traits for that breed. And so you want them to be purebred, that is homozygous, preferably dominant. Those traits are mostly dominant traits that are used for breeding. Because you want to make sure that every time you breed that dog with another dog, that you're always going to get a dog that looks like that, a characteristic animal for that breed. Breeders care about this a lot. And so that's one of the reasons, by the way, if you buy a purebred, they give you a certificate. Ooh, I have a certificate. And that essentially means that they're certifying that your dog, or I suppose cat, I've never heard of it done with cats, but it probably is done with cats too. Your dog is guaranteed to have been from two parents that were purebreds, and that therefore your animal is also a purebred, and could be used for breeding as well. So dog breeders care about that. So, and it's not just for dogs, obviously. I mean, you could breed any animal like that. Horses were the traditional animal that were bred that way. Purebred dogs is a relatively recent phenomenon, but horses have been bred like that for a very, very long time. So, anyway, that's the idea here, okay? So in order to determine whether or not you have a purebred dog, the strategy that historically was used is called doing a hybrid test cross, okay? A monohybrid test cross. Often just abbreviated as test cross. Okay, I'm just gonna call it a test cross from now on, pretty much. The textbook does that too. So, in more modern times, okay, so I mean in the last 25, 30 years, if you want to determine whether or not you have a purebred, you can just get a DNA sample from your dog and just send it in. And someone can run the numbers on it. They can genotype your dog and just tell you, yep, this is a purebred dog for these traits. Oh, it's not a purebred for these traits, or whatever. You can just genotype your dog. But that is a very recent development. of the last 20 years. And I realize that that's, during your lifetime, but it's not that long that we've been able to do that. And before we were able to do that, we needed another method in order to test whether or not an animal is a purebred to use for breeding purposes. This is important for breeders. And obviously, this is not necessarily the most handy thing to know in terms of useful knowledge, because are you going to do this? No, you're not going to do this. There's no point to doing this anymore, because you can just have this genetic test done. And also it takes a long time, and it's very labor intensive. But understanding the process of this, how it was done and why it was done, is important, because it tells you something about how genes are expressed. Okay, which is the reason we still talk about test crosses, even though you would never do this anymore. So let's talk about it then. So I want you to imagine a scenario here where we have two dogs, okay? And we want a black dog, okay? What we want here is to breed some black dogs, and we want purebred black fur, okay? But there are two options in these dogs, two alleles that are present. There is the dominant black, and there is the recessive blonde, okay? We have two dogs, and we want to figure out, one of them is a purebred and one of them's a hybrid. A hybrid, by the way, means a heterozygote. It means it has a recessive allele in it somewhere, okay? And we want to know which of these is a purebred that we can use to breed dogs, and which one of them is the hybrid, okay? So keep in mind that I'm telling you here at the outset which one is a purebred and which one is a hybrid. However, you don't know this before you do the test cross. So the only thing that you actually do know about these two is that they're both black, okay? So they both have a black coat. In other words, both of the dogs are showing the dominant trait. Now, if you had a blonde dog sitting in front of you, well, you'd know it wasn't a purebred for the dominant trait, because that would be the only way that you would know the genotype, right? It would be little b for sure if it was blonde. So you wouldn't have to do the test cross. It's not a good dog for breeding. You already know that. So the reason you do the test cross is because it looks like this dog is a good dog for breeding, but you don't know. You don't know if it's homozygous dominant, and you need a homozygous dominant, a purebred, for breeding. Okay, so we want to determine, do we have a purebred dog? So how you do that, we're going to do what's known as a test cross. I'm going to explain all the stuff that's written up here by actually showing you, by doing it with you, okay? So instead of just reading through that, I'm going to do it with you. So what we have to do, the strategy for doing a test cross, is to cross the animal, in this case it's a dog, with a known homozygous recessive. And we know that a dog is homozygous recessive, we already mentioned this, if it has blonde fur, okay? So you always, in a test cross, are going to cross with homozygous recessive. In this particular case, that means little b. But anytime you do a test cross, you're going to find the recessive trait in an animal. It's obviously showing the recessive trait, and you're going to cross the animal with a recessive animal. Okay? So when some of these practice problems ask you to do a test cross, you're always crossing it with a homozygous recessive individual. That is how you do a test cross. Okay, there's a question on the quiz about that as well. So I wrote the let statements out here again. You would normally write them, right, as part of your problem solving. Remember that dominant allele for black here is capital B, and small b represents the recessive allele for blonde. All right? So we're going to take both of our dogs. I gave them names. I call them Buck and Betsy. Doesn't matter. You can call them whatever you want. Which are black, but they have an unknown genotype. Okay, so that's this right here. Remember, B space or B blank means unknown. We don't know what the second allele is. We know they have the black allele for sure because they're showing the dominant trait, but we don't know the other one. Okay? And you always cross it with a homozygous recessive, which would be blonde in this case. All right? So we cross it with the blonde. So here are the two outcomes that you can get based on what your original dog was. Remember that when I say the original dog, I mean the mystery dog, the dog that we're trying to figure out here. So it'd be either Buck or Betsy. Okay, so let's assume that the dog was an actual purebred. Okay, so this is the Buck scenario. I made Buck my purebred. Okay, so Buck is big B, big B. Well, if I cross big B with little b, which is the blonde, 100% of the time, every single time, I'm going to get a black dog. Okay? You could breed those two dogs together 100 times. You will only get black dogs every single time ever. Okay? So, if you do the breeding experiment and you always get black, that's a pretty good indicator that you have a purebred dog. Okay? If your dog, and I used Betsy as my example of a hybrid, okay, so not a purebred, so hybrid would be big B, little b. If I breed my hybrid here with little b, 50% of the time, and you can look at the Punnett Square here, solve out the Punnett Square, half of the time you're going to get a black dog, and half of the time you're going to get a blonde dog. So 50% of the time it's black, and 50% of the time it's blonde. So if you get any blonde dogs at all in a litter, in your test cross litter, you don't have a purebred. Okay? And so then you figured out that your original dog that you were testing is in fact not a purebred dog. And that's obvious. That's important. And you don't want to use it for breeding. So, to conclude, if all of the puppies are black, you can conclude that your dog was probably a purebred. And this is actually important here. I added the word probably in here. There's a little bit of uncertainty because remember that this is random. This process is completely random. So if I were to take Betsy here, remember that every time I breed Betsy together with that little b dog, whoever that is, it's going to be random which one of these we get. So sometimes it's this one, sometimes it's this one, sometimes it's this one, sometimes this one, and then maybe this one again, and now it's this one, and then maybe this one, and this one, and then this one, this one, this one, this one. It's going to be random which one you pick every time you get a dog. Okay, it's totally random every single time. What that means is, if you have a litter of 4 puppies, it's possible that just due to random chance, you only get these. Okay, it's like flipping a coin 4 times and getting heads 4 times in a row. Okay, I can look up the probability of that pretty quickly, but if anybody's taken finite or whatever it's called now, the senior math course where you do this, probabilities, anybody taking that right now? It exists. That would be 0.5, which is 50% chance, to the 4th power. Okay, so that's a 6.25% chance. So the chance of getting only black 4 times in a row, or flipping heads 4 times in a row on a coin, there's a 6.25% chance that you would do that. Just due to random chance, you just flip 4 heads in a row. Now, the more puppies you have, the less likely it is to get that accidentally all in a row. So let's say that you had 8 puppies in a row. Okay? 8 puppies would be a 0.5% chance. And I'm not going to teach the entire probabilities course here in 2 seconds, but essentially it's 0.5 times 0.5 times 0.5 8 times. Okay? So that's 0.5 to the 8th power. Okay? So 0.5 to the 8th power is 0.039%. Did I get that right? No. This would be, it's 0.39%. Okay? So 0.39% of the time, or 39 times in 1,000, you're going to get, is that correct? 39 times in 1,000? 39 in 100 would be, no, 39 and 10,000. 39 and 10,000 are going to be times where you roll heads, or you flip heads 8 times in a row. Okay, so that's if you have 8 puppies, you have a 0.39% chance of getting all black, which is extremely small, obviously. So it's very unlikely that that's going to happen. So the more puppies you have, the more likely it is that you aren't going to get that. And I better plug my iPad in before it dies here. Anyway, basically, if you get all black, it's probably purebred. Now, on the other side, if you get any blonde at all, so if some of the puppies, it could just be one. Oh, shoot, that wasn't the one I meant to highlight. If some of the puppies are black and some were blonde, the some were blonde is the most important part here. If some of the puppies were blonde, then you definitely have a hybrid. Because no matter what, in this scenario where you have a purebred cannot make a homozygous recessive baby. It's impossible for it to happen in this situation. So you'll never ever get a blonde puppy with a purebred. So if you see any blonde, if you see any of the recessive trait, then you don't have a purebred. Okay? So that strategy is called a test cross. And then again, just to summarize here, you're going to cross an individual of an unknown genotype. Remember, that's what you're trying to figure out. You have an unknown dog or whatever animal showing the dominant phenotype. Then you're going to cross it with a homozygous recessive individual to figure out what the unknown is. Okay? If all of the offspring show the dominant, it's probably a purebred. And if any of them at all are the recessive, show the recessive, then you know that you got a heterozygote for your unknown. All right, so that's a test cross. There is at least one practice problem where you're going to do a test cross, or at least use the idea of a test cross to figure out whether or not something is a purebred or not. Okay, so there are some questions that go along with this. They are analysis questions on page 191. It's number 1 and 2. The first question, number 1A, you're going to use that full 6 or 7 step strategy. Let statement, genotype of parent, phenotype of parent, Punnett Square, genotypic ratio, phenotypic. So that's the first part. Then in B, for part number 1B, you're actually going to work backwards. So it tells you what some of the offspring are. And then you got to do some thinking and work your way back up and figure out, well, what does the parent have to be then in order to make that baby? Okay? And then in number 2, you go back to using the full 6-step strategy again. Okay? So this is just some practice problems where you're going to solve your own genetic problem. Okay, on page 191. Now, so I got to work out how to break the time out today. Now, I could give you the time now to work on these and come back and finish the other lesson after, but I think it's easier if I just do the next lesson now and then just give you all the work and then let you just go off and finish it all on your own. I think that's probably the best way to organize this time. It seems like we're going extremely quickly when I'm doing that, but I think it's easiest for you, especially if you want to take a break. You want to walk away, come back. It's a little harder to do that if you're supposed to be working in between. So I have one more topic that I'm going to cover today, and that's this idea that not every trait is dominant recessive, or what we called earlier, complete dominance. Remember, complete dominance is where there is a dominant trait and a recessive trait, and the dominant always shows if it's present, and the recessive only shows when you have 2 of the recessive. But other than that, it never shows. Okay, so that's called complete dominance. And everything that Mendel worked with exhibited complete dominance. But that is not the only relationship between alleles in nature. Not everything is complete dominance. So I'm going to talk about 2 other types of inheritance patterns. Okay, those are called inheritance patterns, or variations in heredity. This is chapter 5.2 in your textbook, by the way. And then we're going to do some practice problems associated with those variations as well. And then tomorrow we're going to talk about one last variation in heredity. It's called multiple allelic inheritance. So that's the last one that we're going to talk about in the course. And then we're pretty much done, by the way. Genetics. Woo! Anyway. So this is what we've been talking about previously, the idea of the dominant and the recessive. That is called complete dominance. All right? So I want to talk about some other things. The first one that I'm going to discuss here is the idea of incomplete dominance. And this is where there are two alleles for something, but one allele is not completely dominant over the other. In other words, when you have both alleles, you have the hybrid, both of them kind of show through a little bit. And you get a mix in the hybrid. Remember, the hybrid is the heterozygote where you have both alleles. The hybrid shows a new trait that is sort of a mix of the other two traits. That's incomplete dominance. Okay, or sometimes it's referred to as a blend. Okay, so you get a blending of the two other traits. In other words, neither of the alleles is truly dominant in this case. I got way too much highlighting going on here. I got to chill out on my highlighting. So that's known as incomplete dominance. And the most important thing that you need to remember here is you can identify it because there are three phenotypes. Normally there's only two, right? There's the dominant phenotype and the recessive phenotype, and that's it. That's for complete dominance. But in incomplete dominance, there's the dominant, there's the recessive, and then there's the one where you have both. And that is a blend, the one that you have both. So it's a blend of the two homozygotes. So I'm going to give you an example here of a trait that follows incomplete dominance. This is a real one. Okay, some of the examples from the textbook and the practice problems are not real ones, but this is an actual real one. Snapdragons, beautiful flowers. Let me show you what they look like. It helps to have something envisioned in your mind. This is a snapdragon right here. There's a red one. Okay, let me find a white one too, so for comparison, because we're going to talk about red and white. This is a white snapdragon. Okay, pretty flower, isn't it? If you take a white snapdragon and a red snapdragon and you breed them together, by what we've learned so far, you're either only going to get white snapdragons, or you're only going to get red, or some are going to be white and some are going to be red, right? I mean, that's what we've learned so far, is that there's only two options. You can either show the dominant or you can show the recessive. But what actually happens in real life is that if you combine red snapdragons and white snapdragons together, you get pink snapdragons. So pink snapdragons are a hybrid, a third phenotype, so a new physical characteristic that comes from the hybrid. The hybrid actually is a brand new characteristic that is a blend of the other two. Okay, so that does not follow the rules that we've discussed previously. So let's talk about how that works. So first of all, we have a different system of nomenclature because you can't just use capital and small letters. You know, typically we would say, okay, well, you know, red would be small letter and white would be big letter, because they're not dominant and recessive. They don't have a capital and small letter relationship to each other. So we need a different nomenclature. We need like a different way to write that. This is not a good way to write this type of relationship. So what we do instead is we pick a letter, okay, a base letter for the trait. So because snapdragons, we're looking at the color, I'm going to pick C as my base letter, and it's a capital letter, okay? So you use a base letter that's a capital letter, and then you use a superscript to tell which of the alleles it is. Okay, so for color, you can either have color red, in which case you're going to have an R superscript for red, or you're going to be color white, and then you're going to show the W superscript for white. Now you'll notice that when I'm writing these superscripts, and for my base letters here, they are both uppercase. When you're doing incomplete dominance, like we're talking about right now, all of the letters are uppercase. Okay, so it's uppercase base letter, uppercase superscript. All right, so that is the way that you write out that nomenclature. Okay, I mentioned here, recognize this new allelic notation. It's a brand new notation. We haven't used that before, and you always use this notation with incomplete dominance. All right, so here we go. What that means is you now have 3 let statements, because not only do you need to tell me what it will be for the purebreds, for the homozygotes, but you also have to tell me what happens when you get a heterozygote. So in this case, red snapdragons, okay, have homozygous red. Not dominant, because there is no dominant here and no recessive. Everything's dominant. So we would just say homozygous red. So CR would mean that both alleles are red. Okay, that's going to produce a red snapdragon. And I wrote here in my let statement that this is incompletely dominant. If you use capital letters for your superscripts, then it's obvious that already is denoting incomplete dominance. So you don't have to say it twice. You show it with your superscripts. Okay. One thing to note here, and this is a little bit of a pain. The textbook doesn't follow these rules always. So sometimes, it uses capital letters for the superscripts, but it always uses capital letters for the superscripts, even when it's not supposed to. So it's a little bit confusing. The next thing that we're going to talk about, which is codominance, which is another type of relationship. It's similar to this. You're not supposed to use capital lettered superscripts. You're supposed to use lowercase superscripts. But the textbook has decided that it's always going to use uppercase superscripts no matter what. So just watch out for that in the textbook. You can't identify this by their notation because whoever wrote the textbook was not aware that this is not the same notation. So error in the text. Just be aware of that. That's not meant to indicate anything in the text for you. They just apparently didn't know that when they wrote the textbook. That's fine. Anyway, CR is red. White is also homozygous, but it's homozygous white. So how do I actually show white? Maybe I'll do gray like this, and that means white. Okay. So we got red and white, and then the last genotype here is for pink, and pink is the hybrid. It's the heterozygote, where you have one of each. And so when you have red and white together, that's going to give you pink phenotype. Okay? So remember that you identify an incompletely dominant trait because it has 3 phenotypes. And the third phenotype, in this case, the last one here is the third, the hybrid, is a blend. You're looking for, is this third thing a blend of the other two? Okay? If it's a single trait that's a blend of the other two, that's incomplete dominance. Okay, and you can do the exact same problem solving with this as you can with complete dominance, but you're just adding in basically an extra complicating factor here, which is, and I think, are we going to do a problem together? We should probably do one of these problems together. Oh, I do a cow problem later. I'll do this cow problem for codominance. That's okay. One is good enough, I think. If not, people are like, I have no idea what's going on. Then we'll do a survey. But anyway, so why does CR produce red flowers? Well, if you think about it at the cellular level, at the cellular molecular biological level, both alleles are producing red pigment in the flower, right? Remember that an allele is just a gene that is turned into a protein, just like any gene, okay? And that protein is red. It's producing a red protein. If both alleles, both copies of the gene, are red, then the flower is red. in the case of CWCW, both of the alleles say white pigment. Make the pigment white. And so the flower is white. If the organism has CRCW, each cell is going to produce some white pigment from the one gene, and it's going to produce some red pigment from the other gene. And when you mix those two pigments together, you get pink. A brand new color in the end. Okay? That's the CRCW. You've mixed the two together and made a blend. So that's called incomplete dominance, when you don't have an obvious dominant and a recessive allele. Does anybody have any questions about this relationship, or like how you write the relationship? Before we move on, because I'm going to give one more type of relationship that is kind of similar to this. We'll do an example together of a problem that would include that. And then I'm going to just let you loose on some genetics problems and try some of these for yourself. I'm going to keep going. I always jump the gun on this, but if a question does pop up in the comments, I will absolutely circle back. Okay, so another type of relationship in genetics is called codominance. And at first glance, this looks exactly the same as incomplete dominance. In fact, I would say students have a great deal of difficulty telling these two apart. But the key is that there are still three phenotypes, okay? So there is the homozygous phenotype for one allele, the homozygous phenotype for the other allele, and then when you have both, the hybrid, okay? But the key is that in the heterozygote, instead of getting a blend of the two traits, what you get is both of the two traits. Okay? That is what ends up showing up in the organism, is that you see both options at the same time. In other words, some cells are going to follow the one, and some cells are going to follow the other. So what you end up seeing in the organism is both things. And I'll show, if this last example here, this is with the snapdragons, was codominance and not incomplete dominance. It is incomplete dominance in real life, but if it was codominance, what you would see is red, white, and then the codominant one would be red and white. In other words, some petals would be red and some petals would be white, or some parts of the petal would be red and some parts would be white. But you wouldn't see a blending where every cell is both. You would see some have chosen one and some have chosen the other. Okay? So some have chosen one and some have chosen the other is called codominance. So codominance is a little bit different than incomplete dominance in that when you have both alleles, you get some cells choose one and some cells choose the other. And so what the end product is that you get both. Both traits, both homozygous traits showing at the same time in different places. Okay? Again, I know this sounds similar. So when you write this out, again, we need a slightly different notation. I've written here again that there is a new notation for this. And that notation for codominance is, again, you use an uppercase letter for the base letter. That stays the same. But for the superscript, now you're going to use a lowercase superscript. Now I picked W's here, which is rather a bad one to choose because uppercase and lowercase W look different. But it's a lowercase superscript for codominance. Okay, for incomplete dominance, they are both uppercase, and for codominance, the superscript is lowercase. All right? That's how you recognize them from the script, but keep in mind that the textbook completely ignores this, which is infuriating, but I mean, the textbook is what it is. So it doesn't actually follow that, although this is a known notation here for this. So anyway, let me give you an example. And this is another real example, by the way. Red cows, they are homozygous for red hair. Okay, so I've denoted that as capital H, small r. Okay, that's my shorthand here for writing that allele. A base letter, which I picked H for hair, but it doesn't really matter, but it's got to be a capital letter. Okay, so hair. And then the R is representing the specific color, the allele. So capital H and then R for red. Okay? So when you're homozygous for that trait, you have red hair. It's a red-haired cow. If you are homozygous for the other allele, which is white, and I intentionally, by the way, picked two examples of red and white, because it's two ways that you can mix red and white together. So you can either have a red and white blend, like the snapdragons, or you can have a red and white at the same time, like the cows, which is codominance. So I intentionally made them both red and white, but this is the example that's in the textbook too. It's a great example. White here is homozygous white. Okay, so HW makes a white cow. The hybrid, which is HRHW, is a cow, gives the cow fur color called roan. Now, roan is not pink. It's not a pink cow. Okay? That is not a thing. What roan is a mix of red and white. Okay? In other words, some hairs are red and some hairs are white. So it is not a third thing that's a blend. There's no blend. It's just both things at the same time. That's codominance. Codominance is both things being shown. So some cells are choosing red and some cells are choosing white. That's how you get both. Okay? So we've got red hair for one. We've got, oh great, of course this is when my iPad's going to start conking out again. We've got white hair for one. I'm going to have to highlight this in order for you to see it. No. There we go. So we've got red hair for one, white hair for the other. These are for the homozygous traits. And then we have the heterozygote is both. Both things happening, as in some cells, some hair follicle cells are making red hair, and some hair follicle cells are making white hair. So when you combine white hair and red hair, you get roan. Roan just means that some of the hairs are white and some of the hairs are red, and it's a mix all through the cow. But it's not pink. Each cell is not doing both at the same time and blending them. Cells are choosing one or the other. Okay? So in codominance, the cells choose. Now, it's not a conscious decision, but it is a choice. The end product here is that the cells are choosing. Okay, so then if you're looking at why this happens, well, HR produces red because every cell is choosing red. Okay, every cell is choosing to express the red gene and making the red pigment. For white, both alleles code for white, and so obviously every cell is going to be choosing produce white pigment. But in HRHW, some cells, and I should write that. I don't know why I didn't write this. Some cells. Can you see this? This isn't even on the screen. Sorry. Some cells whoa. I think that my iPad is just giving up the ghost here. Hello? Oh. I'm going to restart this. This is too slow. This is unusable now. Hate it when that happens. Okay, some cells express the white allele, and some express the red allele. So there are red hairs and white hairs. Okay, so in codominant relationship, both are expressed. Okay, so let's look at an example here of crossing a white cow and a roan cow. Now, this is like a little tiny example off to the side. I can barely read that. So I'm just going to just pop it onto the next page down here and do it together with you. So I'm actually going to just totally delete this here. This is actually for tomorrow. We won't worry about this. This is actually one of the homework questions. I'll let you do that, and we can even take this homework question up tomorrow. Some people had trouble with this homework question in the other class. If we are in this class, we'll absolutely take it up tomorrow at the beginning of tomorrow's day. But I'm gonna do this roan example here. Okay, so I want you to imagine this scenario where we are going to cross a red cow. Don't know how to spell the word cow anymore. Cross a red cow with a white cow, and what are the genotypic ratios and phenotypic ratios of the offspring? Okay, just like a potential sample question here. Okay, so first of all, we're going to need let statements. I'm going to borrow my let statements from above and say, okay, what did I use above? CW. Yeah, okay, sure. Again, I don't love W, but we'll go along with it. We're saying CW, CW, and CW is white. CR and CR is red. Note, by the way, that these, whoopsie-daisy, these right here are small letters, which implies codominance. Okay. It doesn't do this in the textbook, which is annoying, but when you're doing your own notation, you should probably follow along here that this implies codominance. Instead of incomplete dominance. And then we've got our hybrid potential here, which is C, W, CR. You can put them in any order, and this gives you roan. Okay, roan, remember, is some red and some white. And we're going to cross a white and red. So we need to write down the genotypes of the parents. That's step one. I may as well number my steps over. Or actually, step one was the let statements. Genotypes of the parents. So we've got white. Ooh, that's a terrible W, sorry. White being crossed with red. And then we also need to say the phenotypes of our parents. What we're trying to do here is find the F1 cross, the first filial generation. And this right here is the P generation, or the P1 generation, the parent generation. So this would be white, and it's going to be crossed with red. Okay, or red fur. Then we're going to do our Punnett squares. Nope, not going to do it. Why did you do that? Okay, so one goes at the top. CR, CR, CW, CW. And then let's look at our potential offspring here. So we're going to have CW, CR, CW, CR, CW, CR, CW, CR. Okay, so there's our Punnett square. If we were to write the genotypic ratios here, just step 5. Sure, genotypic ratios. I'm short forming genotypic, by the way, to just gen here, but for brevity. Genotypic ratios are they're all the same. So this is going to be 100% CR/CW. And then our phenotypic ratios are going to be, CR/CW is Roan. So this is 100% Roan babies. So I want to compare this to the exact, remember we did this experiment with the peas, right? We took 2 purebreds, purebred green and purebred yellow, and we mixed them. We got the same result, right? Except for the fact that it wasn't a third phenotype. It was one of the original phenotypes, which was that they all turn out green, right? So in this case, we're getting a third phenotype. That's already something weird. Mendel did not come across this because he didn't discover codominance, but this exists. So what I want to show you is the difference between this and now what happens if we take the F1 generation. Remember, the F1 is just taking the children, the first filial generation from the parents, and crossing them. So that's called an F1 cross. So let's do an F1 cross, okay? We're going to cross the babies from the original union. I'm going to go through all the steps over again here, but I'm going to cross the babies. All right, so we don't need to do therefore let statements, rather, because they're already done. So I'm going to start with my genotypes of parents. CW, CR, crossed with CW, CR. So this would be 2 Roan, right? So step 3 here would be Roan times Roan. Set up our Punnett square. CWCR, that's the one potential parent, and the other parent is CWCR as well. Okay, so we're just putting their genotypes, one on the top and one on the side, and this is how they're going to combine together. So this is CWCW, CWCR, CWCR, and CRCR. Okay, so when we look at our genotypic ratios here, we've got 1 CWCW to 2 CWCR to 1 CRCR. And so our phenotypic ratios, that's what it physically looks like, are going to be 1, this is also going to be 1 to 2 to 1 of white to Roan to red. Okay, and notice how that is different from the complete dominance example. We did the exact same cross for complete dominance, but we saw 3 to 1 dominant to recessive. 3 greens to every 1 yellow. If you remember way back when we did this with complete dominance. So you'll notice that the actual results that you get at the end of the experiment look different. Okay. So you'll notice, by the way, that this doesn't change anything about how you do the problem solving. It just changes how you interpret the results, because you have to interpret them by sort of like a new let statement system here with a little bit different notation. But other than that, how you actually do the problem solving is exactly the same. Okay, so what are you going to do with all this? Let's get to what you're working on. So you, man, we ever cover a lot of stuff in this course. It's crazy. Okay, so yeah, and if you have an opportunity, watch the heredity video. It's not absolutely essential, although it does give you a little bit more background on Mendel and how these theories were developed. I suppose you could consider it enrichment, but it is good to know. None of it's directly in the quiz. All the stuff that we covered, the quizzes are built off the stuff that we cover directly for the most part. There are some key videos, but I always tell you what they are. Exit quiz. Try it out. At least see what your understanding is like for basic inheritance, understanding the idea of dominant and recessive, and when you show the dominant and when you show the recessive. That's what that little quiz is about. Okay? Then you are going to complete question 5 on page 193. Some of you have done that already. You're going to try on page 199. These are the practice problems that I already mentioned. It's numbers 1 and 2, okay, at the bottom of 191. And then I have 2 practice quizzes for you to try. Okay, again, these are optional, but I encourage you to try them and test out your skill. One of them is on just inheriting a single trait, okay, which is basically just using a Punnett Square and doing complete dominance. And then the other part here is on just using Punnett Squares and test crosses. What is your understanding of how to use a test cross and what the results are that you see when you do a test cross? Okay, then there is a supplementary video here that gives some examples of incomplete dominance and codominance. And it talks a little bit about the idea of polygenic traits, which we're only covering Mendelian traits in this course, but it talks a little bit about how polygenic traits work. So if you're interested in that, you can watch that. If you are going to watch this video, the only part that's important is the part where it discusses the difference between incomplete and complete dominance. A lot of people have difficulty telling the two apart. And so this one gives you some sort of maybe like an additional way to think about it, about incomplete and codominance. And so I recommend it for that reason. The second part that's on epistasis and polygenic traits, you don't need to know that for this course. So once you get to that spot in the video, you can just stop. And then there are some practice questions. These questions are on incomplete and codominance. It is page 196, 1 to 3, and number 5. And there is an exit quiz to practice that stuff as well. The last part here on blood types and multiple alleles, we're going to cover tomorrow morning. Obviously, we're out of time for it today. And that will be our last topic in genetics, other than to work on the project. Oh, actually, that's not exactly true. We're going to take a few minutes to do a reading on genetic illnesses. That's the other thing that we're going to cover tomorrow morning, but that's relatively short. Any questions before I send you off to work on this stuff? You likely will have questions after you work on the practice problems, just as a heads up. And so if you do, you can either drop in during the Google Meet, but please know that I would be happy to take them up at the beginning of next class because most people will probably want me to. So if you come in and have me do it with you in the Google Meet, which is totally fine, just be aware that I might do it twice. What does it mean when a site says that an animal has 6 or 7? Ah, you're looking for the project. That means that males have 6 and females have 7. Don't pick a species like that. They have a different way of establishing what is male and female. They basically just don't have a Y chromosome. Don't do a species like that. The actual way that works is fairly complicated and is going to make your project far too difficult. So just don't use that species. I mean, I guess you could use the species and just say it has 6. I mean, you could do that, but I wouldn't. I just wouldn't use it. like I said earlier, I probably say this throughout the course. Biology is all about being like, oh, hey, here's also a bunch of rules about how things work. And then you spend like 90% of your time talking about the exceptions to the rules. Chemistry is like that too. And so that's just an example of an exception. There's a lot of them. Any other questions about this content? Actually, you can go to anything. It doesn't have to be with this content. But all right, I'm going to let you guys go to work. And again, I'll be here the whole time. So just let me know if you need anything, and I'll be here in the Google Meet. Drop by if you're confused about a particular topic, and we will absolutely dig into it together. All right. Good luck.